Good morning and welcome to the Targeted Therapies Forum 2025. It is an honor to welcome you to this milestone event, the 10th annual Targeted Therapies Forum for Grace. So happy anniversary to us. Over the past decades, this forum has served as a virtual platform for advancing knowledge of precision medicine in lung cancer. Today, I'm joined by a panel of leading lung cancer experts, including medical oncologists, radiation oncologists, and researchers who are in the forefront of developing and implementing targeted therapies. Throughout the day, we will explore multiple aspects of targeted therapy in lung cancer, ranging from the fundamentals of cancer-specific mutations to in-depth discussion on the latest advancement in managing each molecular subtype. Our goal is to provide both foundational knowledge and cutting-edge insights, ensuring that you leave with a deeper understanding on how to optimize care for patients with oncogen-driven lung cancer. We encourage you to engage and ask questions at the end of each panel presentation. And with that, let's get started. Welcome again, and thank you for being a part of Targeted Therapies Forum 2025. It is my pleasure to introduce our first speaker, Dr. Triparna Sen, who is an associate professor of oncological sciences and director of the lung cancer PDX platform at the Icahn School of Medicine at Mount Sinai, New York. Her groundbreaking research focuses on the biology of lung cancer initiation, progression, and drug resistance, uncovering therapeutic vulnerabilities and advanced treatment outcomes. Welcome, Dr. Sen.
Thank you so much, Dr. Bezanova, for that very kind introduction. Good morning, everyone. I'll try and share my screen. Well, I'm going to talk about, since the privilege of being the first speaker in this webinar, I'll try to break down some of the concepts that I'm sure my esteemed colleagues are going to walk you through for the rest of the day. I am a translational researcher, so I work in the lab. I have a lab at Mount Sinai, and my lab is really interested in understanding the biology of lung cancer so that we can have better therapies, and we can also choose patients who will best fit that therapy. So with that in mind, my talk is about bench to bedside. What I thought I would do today is really go to the basics of what is cancer research, what does bench to bedside even mean? What is DNA, RNA, and protein? What is in vitro and in vivo characterization? When you pick up a paper and you read about in vitro and in vivo, what does that really mean? And what are these high throughput technologies and analysis tools that has helped us understand so much about cancer in general, and of course, lung cancer, which is our topic of discussion today. And then I'll touch upon a little bit about the discoveries from the Sen Lab. This will be mainly towards small cell lung cancer and also non-small cell lung cancer. So with that, let's get started. So what is cancer? So cancer, I think, we have to acknowledge that it is a very, very complex and a heterogeneous disease. It is characterized by the development of the normal cells in the body that divide uncontrollably and they refuse to die. So it's really your normal cells that refuse to die. And the cancer cells have the ability to infiltrate and destroy normal tissue, and that's what makes them so fatal and leads to the morbidity and mortality for this cancer. And to make matters worse, cancer often has the ability to spread to other parts of the body through the blood or the lymphatic vessels, and this is what we call metastasis. So just, thinking about cancer, and that's why the cancer research takes so long, is because it's a complex disease. It's not one disease, and every patient is actually very different from the other. So there is a long time span of what we consider drug discovery and development pipeline. It often takes anywhere between 8 to 20 years. And it starts with these processes of identifying a good target and then having, let me see if I can never mind. It starts with the target identification. It goes to, having target assays and constructing your best sort of targets. And then we go to validation of these targets, and then it goes to the clinical phase. My work and sort of the talk for today will concentrate mainly on what happens before we go into the clinical setting and the validation of these targets, which often takes years. 2 to 5 years is the lifespan of what we can spend in the lab in order to really come up with a very good target and take it to the clinic. So what is cancer research? I told you about cancer, but what is cancer research? Cancer research is also quite complex, where different components come together. So the basic research, which we do in the lab with cell lines, with mouse models, and then translational research is when we try to take this finding of the biology from the lab to the clinic. Clinical research is when you take human tissue and other things from the clinic, and then you try to study biomarkers and other things. And then population research is to understand more epidemiology, the risk factors that predispose a certain individual towards a cancer risk. So these are all very important components of cancer research. What my work has mainly concentrated on is basic research and translational research. And why do we need cancer research? I think drug discovery, not only is it a very lengthy process, it's also a very expensive process. And this is a study which kind of talked about, what is the degree of success when you go to a phase 1 or a phase 2 clinical trial, a phase 2 or a phase 3, and then phase 3 to approval. And you will see that depending upon, what disease you are in, oncology is right at the very beginning, the degree of can be quite low, right? Because we have to develop efficient tools, we have to come up with the speed, we have to come up with reliable targets, we have to be efficient, and we have to choose the best patient for the best drug. So in order to make this process as efficient, reliable, and speedy, we need cancer research. We need to learn what we understand about the biology in order to ensure success in the clinic. So the goal is to move basic science discoveries more quickly and more efficiently into practice. And that is why cancer research is sort of our true north, which we have to do in order to have more effective therapies in the clinic. And where we are today is a true testament of what cancer research can do, because cancer research has accelerated lung cancer drug discovery and approval process. And this is a picture from the Lung Cancer Foundation of America website, Lung Cancer Research Foundation website. And as you can see, going back from the early 90s, now we are at the 2020s, we have seen in leaps and bounds, where we are at with the approvals and with pushing drugs from the clinic, from the lab to the clinic. So the targeted therapies, immunotherapy, chemotherapy, we are at a really exciting time of cancer research where we are seeing real movements in the clinic. And I think for the longest time, we couldn't associate lung cancer with long-term survivors. And now we are seeing long-term survivors in lung cancer. So I think thanks to cancer research, which has really accelerated this drug discovery and approval process. So I thought, we talk about a lot about DNA, RNA, protein. I really wanted to break it down to what that really means. So if you think about DNA is really like a master instruction book for life. It contains all the information and to run the body. So it is an instruction manual. Think of it as like a recipe book, which contains all the recipes, which are your genes, that tells your body, that tells your cells what to do. So that's your DNA. It's an instruction manual for life. The RNA is really the messenger. So it's sort of one recipe from that cookbook that your body needs or your cells needs at that time to make the protein. And I'll tell you what the protein is. So it's really, RNA is like the messenger. And the protein is like the finished product. If DNA is a cookbook and RNA is a copied recipe, then protein is the final meal that you see on your table. It takes the instruction from DNA, and this protein is actually very important because it does all the important functions in your body, like building muscles, digesting food, fighting diseases. So this is what DNA, RNA, and protein is all about: recipe, cookbook, and your final meal. So bench to bedside research is it's no longer unidirectional. In my lab, I have collaborators everywhere in every aspect of research. And I think I talk to them almost every day in order to make my research more timely and better. So we go from the diagnosis and treatment in first line and second line. This is typically where from the patients, it could be blood, it could be other samples, tissue, and other body fluids. And then we build cell lines and mouse models from these, either blood or the tissues. And then we do array of techniques. And I'm going to talk about the utility of each because, there's a cost associated with all of this. So then we take that to what we call preclinical research. This preclinical research then informs the clinical trials. It could be phase 1, phase 2, phase by, several fantastic clinicians who will tell you a lot more about this phase 1, phase 2, and phase 3 trials. And then we learn. It's not unidirectional anymore. We learn from these trials, we go back to the lab, and then we do this process again to make the process more efficient, more better. That's always the goal of translational and basic science. So let's talk about, the cell lines and the mouse models. So, if you've been to conferences and talking about lung cancer, you will come through this slide a lot. How has cell lines and mouse models really helped us in developing better therapies, right? So I think non-small cell lung cancer is a true testament of molecular testing in terms of developing targets and better therapies, right? Now, when you think about lung cancer, it's no longer one disease. We can divide lung cancer into these different subtypes, which are driven by these driver oncogenes that you'll hear about later in the day, like ALK, EGFR, KRAS, and there are drugs against it, which the patient gets when they come into the clinic. And now, thanks to basic research and translational research, we understand so much more about the disease that we are thinking beyond just oncogene, the single oncogene. We are thinking about co-mutations or the alterations in the genes that happen together. And one very good example is the KRAS mutation, along with its two co-alterations like STK11 and KEAP1. So this was a study that came out of Memorial Sloan Kettering Cancer Center, which looked at KRAS mutations and all the other co-mutations that predict poor outcome in this subtype of the disease. And what they found was that in a KRAS mutant tumor, having a co-alteration in these two genes, STK11 and KEAP1, actually predicted the worst survival and the worst prognosis in these patients. So we went back and we looked into these data sets of 1,235 metastatic lung adenocarcinoma cases, and we found indeed in these patients, if you just concentrate on the black line here, the patients who had KRAS mutation along with STK11 and KEAP1, they had a much worse prognosis than the patients who did not have mutations in these genes. So the real question that we asked in the lab is, what is the function of these genes and why do having these co-mutations make this cancer so aggressive? So for that, we use the cell lines in the lab, and that is the utility of cell lines. So we use these two cell lines, H358 and NH292. We actually knocked out or we deleted these genes in these cell lines, and we wanted to study what is the biology, what can we learn from the RNA, from what I told you, what can we learn from these processes called RNA sequencing? I'll talk about RNA sequencing in a bit. And also with CRISPR screens, where we delete different genes in these models, and then we try to identify what happens and what are the phenotypes of these cell lines. And what that led to, I'm not going to go into the details, but what that led to is to understand that there is a specific vulnerability in these cancers, which is called a ferroptosis or a cell death pathway. And there are several genes that are involved in this process, which could be targeted. And right now, there are preclinical studies and also translational studies going on in trying to understand the importance of ferroptosis in this cancer. So this tells us that we can learn a lot from cell lines, and this can actually be taken into the clinic. Mouse models, very important. There are several kinds of mouse models. The work that we do mostly is with patient-derived xenografts and with genetically engineered mouse models, and I'll tell you what these have really taught us. So patient-derived xenograft models is when either blood or tissue is collected from the patients and it's engrafted into a mouse. And when the tumor grows, it's really representative of the tumor that we see in patients. We can do drug studies in these models so that we really understand, because we can't take every drug or target into the clinic, right? We only have to take our best drug and best target into the clinic. So we can do these trials in the lab with these patient-derived xenograft models to understand toxicity, to understand the effectiveness of these drugs in these preclinical models. And genetically engineered mouse models are really, if you have a KRAS mutation, we try to mimic this mutation, like a KRAS mutation or a P53 mutation, into the mouse model so that we can understand the biology of this disease, and we can also understand what are the therapeutic vulnerabilities of that particular cancer type. So, I am also the director of the lung cancer PDX program at Mount Sinai. So we collect CTCs, biopsies, samples, and then we are engraft into these mouse models. And we can do different techniques. We can do histology, we can do whole exome sequencing, proteomics, RNA-seq, single cell sequencing, and drug response studies. And I know you're going like, whoa, 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 what are these techniques? I'm going to talk about them in a little bit and what that has really taught us. But the point is, we can understand a lot about the disease from these mouse models. I'll start with the drug efficacy studies, and you will see a lot of these sort of dose response curves or tumor volume curves in this, any paper that you pick up, right? So this is with the PDX model, which was treated with two drugs, samatolisib and osimertinib, and we could understand that these drugs, the combination of osimertinib with samatolisib, if you look at the red line here and sort of the dark pink line here, if the curve is going down, which means the tumor volume is going down, which means that this drug is actually working in our mouse models. This is another example of that, where we worked with lurbinectedin, again with osimertinib, and we saw that if you see the red line here, that the combination is doing much better than this yellow line, which is osimertinib by itself. So really, mouse studies can really tell us that this drug is actually working with the other drug, either as a combination or even by itself without causing a lot of toxicity in the mouse. So that is the beauty of these mouse models. Again, this is another genetically engineered mouse model where we typically do immunotherapy studies, because these are grown in mouse that have intact immune system. So in this study, again, we found that anti-PD-L1, and this is a small cell lung cancer mouse model, where we found that, with ceralasertib, which is an ATR inhibitor, it works in combination with anti-PD-L1, and we are able to do a lot of downstream studies from these mouse models. And for immunotherapy studies, genetically engineered mouse models are really important. And again, we could compare what we found in the mouse with a clinical trials. So this is a proof of concept trial with berzosertib and with topotecan, and we were able to do RNA sequencing from these tissues. And the point is, what we found in the mouse, what also confirmed in the patient samples. So this tells us that this drug is working in a similar fashion for the most parts. And this study, what that led to is to identify ATR inhibition or DNA damage inhibition as sort of an effective target. I'm not going to go into the detail of the biology, and I'm happy to answer those questions. But what that really, these studies are telling us is this is a drug that could actually work in this disease type, and it works by activating the immune microenvironment. And what that leads is to clinical trials, because that's our real goal, is can we take these drugs from the lab into the clinic? And what that study did is, support a lot of the clinical studies for this class of drugs, the DNA damage response inhibitors with immunotherapy for small cell lung cancer. Again, this is another example where we tried to mimic the maintenance setting with a drug called lurbinectedin, which is currently approved in the second line for small cell lung cancer. And again, this study told us that lurbinectedin could be an effective maintenance strategy for small cell lung cancer. And the beauty is, this is a trial, which is the IMforte trial, which is looking exactly at lurbinectedin with atezolizumab in the maintenance setting. And our study is a preclinical validation that this combination could actually work in the clinic. And there was a press release which told us that this work could be, this combination could be effective for the patients with small cell lung cancer. We still have to see the data, but I think the point is that this is the pipeline that we want to follow, is take a drug from the preclinical setting to the clinical setting. So I talked a lot about RNA sequencing, proteomics, and all of that. What does this really mean? And why do we invest so much of our funding into these high throughput techniques? So I'll talk about histology, what that tells us, immunohistochemistry. So immunohistochemistry is a very basic thing that is done as a part of clinical research and also at preclinical research. What histology or immunohistochemistry tells us, and what this told us from this study, is the morphology of the cells. How does the cancer look under the microscope? What are the markers or the proteins that this cancer expresses, which can be used for diagnosis? And what are the specific markers that, in our case, small cell lung cancer expresses that can tell us a lot about the disease when we try to identify drugs? And it also tells us that this cancer is actually very heterogeneous. When we talk about NGS or whole exome or whole genome sequencing, what that tells us is about the high mutation burden, what are the genetic alterations that can be targeted, what are the genetic alterations that cannot be targeted. And this told us that small cell lung cancer lacks a lot of the recurrent genomic alterations that can be targeted. But for non-small cell lung cancer, these studies told us that there is a KRAS mutation and an EGFR mutation that could be targeted. Proteomics, it again tells you a lot about the proteins, and it tells us about the drugs that can be targeted. And this is a lot of the study that we did, is we found through proteomic analysis that some of the drugs targeting DNA damage response pathway could be effective targets. Finally, RNA sequencing tells us the subgroup of these tumors and also helps us understand some of the targets that are currently in the clinic. You may identify DLL3, you may identify PARP and other targets that are currently into the clinic. Finally, single cell RNA sequencing. Why do we need that? Because we need to understand rare population of cells. And if you consider bulk RNA sequencing like a smoothie, we really want to understand the fruits that went into that smoothie. And this told us a lot about small cell, about the heterogeneity, about the immune microenvironment, and about rare populations. So with that, I'd like to, sort of bring all this together, because I know I took you through like a lot today, but I would really want to break it down, because basic science, we invest years and years of our life into understanding the biology of the disease so that we can take targets into the clinic. And it really transforms cancer care. Breakthroughs happen only when waiters and you, patients and caregivers, we join forces and decode the mysteries of cancer. There's a complexity to the drug development process. It cannot be hurried, because we have to identify new targets, we have to take new molecules, and the deeper understanding of cancer biology will lead to better treatments. Life scientists, we are the partners in innovation, so talk to us. From bold ideas to real world impact, that's what we invest our time in. Researchers drive the future of cancer care, because with every discovery, we hold the potential to change lives, which becomes a therapy for tomorrow. What do we need? We need cutting-edge infrastructure, because it is a foundation of understanding cancer. We need resources, we need funding, we need partnerships, we need perseverance, most of all. Years can go into understanding that. State-of-the-art technology, I spoke about some of them, I'm happy to answer more questions, but it accelerates discovery in an unprecedented space. And then you need brilliant minds to come together. We need to have open conversations like this, where we all come together into finding new therapies. I want to thank everyone, all my team members, but innovation and research saves lives, and the future of cancer treatment is being built today. And with your help, the future looks really promising. With that, I'd like to thank you all, and I'm happy to answer questions in the Q&A session. Thank you.
Thank you so much. Thank you, Dr. Sen. That was wonderful. We'll leave the questions to the Q&A part of the presentation today. And it is my pleasure to introduce our next speaker, Dr. Jennifer Marks, who is a rising star in thoracic medical oncology. She's a thoracic medical oncologist at Dana-Farber Cancer Center Institute and Brigham and Women's Hospital. She's an instructor of medicine at Harvard Medical School. Her research focuses on precision medicine and biomarkers, and she has a special interest in thymic tumors as well as cellular therapies in lung cancer. Go ahead, Dr. Marks.
Thank you for that kind introduction, Dr. Boxkanovich. I will share my screen. Sharing, I believe.
We are seeing it, but we are seeing it in just normal mode, not the presentation mode.
1 second. Let me try this again.
And if we can't have it, we can ask Grace to share, and you just guide us through the slides.
Okay, how about now?
Now, perfect, works. Go ahead.
Okay, sorry for that delay, everyone. Hi there. Thank you so much for that very kind introduction. I'm so excited to be here and to talk to you about biomarker testing in non-small cell lung cancer, or in lung cancer in general. Today I'll be giving you an overview of biomarker testing in lung cancer itself. And we'll cover some of the genetic alterations and testing modalities that Dr. Sen so beautifully laid out in order to orient you to these advances and understanding of precision medicine. So we'll go through a basic introduction to biomarkers. We'll then go on to look at the genetic alterations specifically that we test for in lung cancer. We'll review the testing modalities and further go through who and when should we be testing for these genomic alterations. So taking a step back, what is a biomarker? So a biomarker is really any molecule that can be measured in blood, bodily fluids, or tissues. And so this is a measurable indicator of something oncologists use regularly in order to either determine the treatment, or prognostically it can help give more information about how the cancer might behave. And so in non-small cell lung cancer, this becomes incredibly relevant because of the revolutionary technologies and advances that we've had in cancer care that have driven really the field forward to allow us to be able to provide targeted therapy to our patients. So that's generally what a biomarker is, but what are the different types of alterations that exist? Well, so in non-small cell lung cancer, there are genetic driver alterations. And so when I say genetic, I don't mean that this is something that can necessarily be passed down to your family members. It's really what's called a somatic mutation, or a mutation that we're really looking for that drives the cancer to make more of itself. And so while there can be associated mutations that can be passed down, by and large, we're talking about the actual mutation of the tumor. And so these could include point mutations, which for example, you'll hear about later is a mutation with EGFR or BRAF as an example, specifically V600E. There are also fusions where two genes combine together in order to make an alteration. And this genetic change is what is driving the cancer forward. ALK and ROS1, which we'll hear about later again, are other types of fusions. Moving to other methodologies, we have amplifications, which means that there are more copies of a gene than are normally there. And this can also be in the form of overexpression, which can lead to increased protein growth or activity in the cancer itself. Deletions are also common and can occur as an alteration that drives cancer. So what genetic alterations specifically should be tested for in lung cancer? Well, in truth, this is an ever-expanding list of biomarkers. As Dr. Sen said earlier, while we don't have while we don't have a medication available for every single driver or alteration that we have in lung cancer at the moment, the hope is that we will develop more drugs and move towards further clinical trial development in order to manage some of these and provide more treatment for our patients moving forward. But some of the alterations we have now are actionable, meaning that we have FDA-approved targeted therapies for, while some of these other ones you see listed here may help to stratify patient prognosis. And so truly, based on the ones that we have available now, these include EGFR mutations, ALK fusions, ROS1 fusions, KRAS mutations, NTRK fusions, BRAF V600E mutations, MET exon 14 skipping mutations, RET fusions, HER2 mutations, and now NRG1 fusions. And while this is, not at least this is the one that we have FDA-approved drugs for, PD-L1 expression, which is a measure of a protein on the surface of cells, is really a major driver in helping to aid in treatment choice if a driver mutation or an alteration is not identified that we have a targeted therapy for. This marker helps to identify which patients might benefit from immunotherapy, which is a treatment that utilizes your own immune system to fight the cancer. And so as alluded to before, these genetic changes also are becoming more important to know about upfront when picking treatment choice, even from a prognostic standpoint. And so how do we test for these biomarkers? Well, in short, there are two major ways in which biomarkers can be tested. And first, we'll review here is tissue-based testing. And so this includes a multitude of test modalities, from older, less used methods to methods that are still used in clinical practice, but are with the advancement of new technologies, we have different comprehensive biomarker testing on tissue available. And so this will include single gene testing, which is where we're looking for a single gene at a time on a tissue, or that's in the tissue. So this is either by a sequencing modality called Sanger or PCR. And some of now this is less commonly used based on limitations in efficiency, but it still can give us a fast turnaround time in some modalities such as EGFR, or and/or it is cost effective compared to some of our other more extensive testing modalities. This fluorescence in situ hybridization is another modality that is still used in clinical practice, more in the hematologic setting, though it does hold a place in non-small cell lung cancer. It's basically taking two probes, fluorescently labeled probes, so they light up a certain color, and when a fusion is present, you can see the combination of the colors combined that helps to determine whether or not there's a fusion or a rearrangement present. And so this can be quick and highly specific. However, technically, it can be a more complex way of determining whether or not one of these fusions is present. And it requires a lot of high quality tissue in order to efficiently or completely test for accuracy. As I alluded to before, immunohistochemistry is used for protein expression. PD-L1 is a perfect example, as is ALK or anaplastic large cell kinase, which or lymphoma kinase, which is another method that we can use that's cost effective, but it can use more tissue. And so it may limit modalities of testing that can be done that are more comprehensive and give us a more complete picture of what's going on genetically or molecularly. Next generation sequencing is a simultaneous test where we look at many gene alterations simultaneously or sequentially, depending on the platform. This is looking at DNA and RNA-based sequencing at times. So different structural components from the tumor to determine if one of these alterations that I previously mentioned is present. And so while this testing is some of our most comprehensive and can detect a rare mutation more easily than one other single modality, this leads to a longer turnaround time in order to get that information back, on the order of 3 to 4 weeks at times. And at least the DNA-based next generation sequencing component at times may miss smaller fusions. And so it's really going into these in more detail. DNA-based sequencing is really, there are multiple kinds of ways to detect or to utilize DNA-based sequencing, which is out of the scope of this presentation. But with the assistance of RNA-based testing, it can certainly improve to ensure that we're not missing these fusions and variant alterations that might identify a target that we have for our patients. And so these two modalities really work together in that sometimes DNA-based next-generation sequencing can miss some of these finer or smaller indel deletions or deeply intronic or exonic alterations that are actually present. And so these two types of sequencing go hand in hand to improve the detection rate of the molecular sequencing. It reduces, at least with RNA-based sequencing, it helps to reduce some of the challenges that are imposed by longer or technically difficult DNA-based NGS testing or sequencing of the long introns or repetitive introns, which are sequences within the gene that may be more difficult to identify a target. And so while we have this great comprehensive methodology of detecting these mutations, liquid biopsy or liquid-based testing is a newer modality of testing these treatments in clinical practice, and is another way in which we may be able to test for these biomarkers. And so this has been most done often in lung cancer via circulating tumor DNA, or what's called ctDNA. This is really detecting DNA from the cancer cells that die off and then are released into the bloodstream. And this helps us to detect the potential target within a time frame of approximately a week, as opposed to 3 weeks with our more comprehensive testing. And so this leads to a faster turnaround time if we're able to identify a target. It's a blood test, so it's non-invasive. And you know, there are clinical trials that are up and coming that are looking for us to use this modality as a means of monitoring disease and possibly in the future predicting those patients who are going to be more at risk for relapse. But this is still in clinic or developing in clinical practice. And so comparing the two methodologies, tissue versus liquid-based testing, I think it's most important here to emphasize that while these tests work hand in hand, or that these tests work hand in hand and should not be used alone, they complement each other. Comprehensive molecular testing using DNA and RNA-based next-generation sequencing should be performed. But these, as I said before, these methods take 3 to 4 weeks in terms to get that information back in order to be able to provide a treatment plan at times for patients. Liquid biopsy by ctDNA testing can get us an answer much faster within about a week. But it's important to say that if this if this liquid biopsy testing is negative, we still need as oncologists to look at the tissue as this is the gold standard in order to identify one of these molecular alterations. And so in the age of precision medicine, the sequence of treatment that you'll hear about later is really essential to identify targets if a target's present, because it can have toxicity consequences if we don't pick the right drug at the beginning, in terms of giving immunotherapy before a targeted therapy. So while the testing can take time, it is still important to make sure that we are aware of the molecular profile for the patient. And so who and when should we test? Well, my biased opinion is arguably that we should be testing everyone up front. But in clinical practice and based on guidelines, this isn't always possible. And so per the guidelines, really patients at diagnosis should be tested for EGFR, ALK, ROS, at least in the earlier line settings or early stage disease, as that helps us to identify our treatment plan. But certainly everyone in the advanced setting or metastatic setting or stage 4 setting should be getting comprehensive testing on tissue at a bare minimum, if not both, liquid and tissue-based testing. These are the guidelines by various organizations, as you can see here, as we have readily treatments available in these specific biomarkers or targeted spaces. So my main takeaways here are that there are many and up and coming biomarkers in lung cancer. And there are differences in identifying tissue-based testing or identifying these markers via tissue-based testing versus liquid-based testing. Turnaround time and how quickly you can get the results back is certainly important, and time is of the essence in determining treatment for our patients. There are 9 or greater than 9 targets at least that are that we have FDA approved agents in non-small cell lung cancer. So we should be testing for all of them in non-small cell lung cancer in order to help inform our treatment decisions for our patients. Most, if not all, patients should be tested in my opinion, every stage of lung cancer, as this will help to guide treatment for patients and pick future treatment or clinical trial options should they become available. But there are variations here in clinical practice and by societal guidelines. Thank you very much for your time, and I am happy to take any questions later at the Q&A session. Thank you again.
All right. Thank you, Dr. Marks. And it is my distinct pleasure to introduce my colleague, Dr. Karen Young, who is an assistant clinical professor at UC San Diego Moores Cancer Center, the place that I practice. She's a medical oncologist who specializes in both lung cancers as well as head and neck cancers, and she is a graduate of UCSD Hematology Oncology Fellowship Program, of which I'm a director. And so I'm very proud that we were able to entice Dr. Yoon to stay at UCSD to continue her practice. And her talk would be more of a practical guide as to what do you do when you see your molecular report. And I think it's very important for you as a patient to make sure that you ask for a copy of your molecular report and try to understand which specific alterations your tumor has. Go ahead, Dr. Yoon.
Okay, thank you, Dr. Bajanova, for that kind introduction. So today I'll be talking about how to read a molecular testing report. And To understand, how to interpret the molecular testing report, I think that it's important just to have a little bit of background of what genetics is. And as Dr. Sen had talked about in her talk, as far as what DNA is, that's what encodes the genetic information. And when we're talking about genes, genes are the segments of DNA that contain information for making the proteins. And proteins is what make up all the building blocks of life. And so it's these changes in genes that ultimately can lead to cancer development and growth. And so when we are talking about driver alterations or biomarkers, as Dr. Marks had talked about in her presentation, driver alterations are those changes in the DNA that can cause normal cells to become cancer cells and lead to cancer growth. And as Dr. Marks had talked about in her presentations, there's different driver alterations. So there are gene mutations, which is a change in the nucleotide or the building block of DNA. Gene mutations can be seen, in EGFR mutations, KRAS mutations. Other driver alterations also include gene translocations, also known as gene fusions or rearrangements. And this is when there's an abnormal rearrangement of DNA that connects two genes together. And some of the common gene rearrangements that we can see in lung cancer include ALK, ROS1, NTRK, and RET. And the other type of driver alteration is a gene amplification where there's an extra copy of a gene. So this is, some a chart that Dr. Sen had also included in her talk, but this is just demonstrating how in lung cancer, there are a number of driver alterations now that can be found in lung cancer that can cause lung cancer. And this is just highlighting how these driver alterations exist, and it's important for us to see if our patients harbor any of these alterations because they can impact what therapy we ultimately recommend. And this is just taken from the cancer guidelines from the National Comprehensive Cancer Network, just to really emphasize how it is important to do molecular testing because, it began in the advanced or metastatic setting where we were really utilizing targeted therapy in patients who harbor driver alterations. And as Dr. Marks had mentioned in her talk, we're starting to use targeted therapy in the early line setting. So molecular testing is certainly becoming important across all stages of lung cancer because this really can influence the treatments that we recommend for patients who have lung cancer. And so I'm not going to go into too much detail on this slide, but as Dr. Marks mentioned, there's, as far as obtaining molecular testing, this can be done on the tumor or the tissue or in the blood, also known as a liquid biopsy. So let's get into looking at an example of a molecular testing report on a patient who had a tissue biopsy. So this is an example of a patient who had their molecular testing performed on tissue. So this is a summary page of a patient whose tissue got sent to Tempus for molecular testing. Starting off on the corner here, usually on the summary page, we'll have information about the patient, including their name, date of birth. And then in this section here, there's a section summarizing the tests that were ordered by the physician, as well as the date that was completed. Here, what you can see is that there's a number of tests that were ordered. For example, when we send tests to the company Tempus, the XT refers to the DNA sequencing and the XR refers to the RNA sequencing. And then the bottom part of this summary then shows the overview of what the results are. So here, what we can see is that as far as the biomarkers that came up on this test, they're listed here in the middle of the page. And this patient had an EGFR mutation. And with that, it's shown in this in the summary page, as well as the approved therapies that are available. And we'll go into more detail about the report in the following slides. So we were looking at the summary report, but when we're looking at the molecular testing report, it's many pages. And as you're going through, the molecular testing report, they can become very detailed. So, beyond the summary page, there can be the report for each specific test. So here on the top right corner, you can see here, there's the XT. So this is the molecular testing report for the DNA sequencing. So on the top, it shows that this is the XT report. And then here on the left side, it's also specifying which molecular testing this which specimen this molecular testing was performed on. So it's indicating that this was performed on a right upper lobe lung biopsy. And then usually it'll also specify when that biopsy was performed. Here in the genomic variants section, I would say this is probably the most important part because this is what's highlighting the genomic alterations that were found. So as far as the genomic variants goes, you can see here that in the potentially actionable section, this is listing the alterations that are found on this specific tissue biopsy that have any associated therapeutic, diagnostic, and/or prognostic indications. So here, this patient has an EGFR mutation. On the bottom of that, there's also a list of biologically relevant alterations. So this patient has a biologically relevant RBM10 mutation, but biologically relevant alterations don't have associated therapeutic, diagnostic, or prognostic indications, but they're listed there because they may have biological significance. Going into a little more detail about the actionable mutations that are reported. So when we're looking at the report here, this is indicating that EGFR is the gene that is mutated. But the EGFR is just referring to the gene, and it gets very specific because there are different types of alterations even within the EGFR gene. So after that, what we can see here is that this report is describing the kind of DNA change that we're seeing in the EGFR gene. So here it's indicating that it's an L858R mutation in the EGFR gene. And here on the right side, the report also mentions the variant allele fraction, which is the percentage of this variant mutation for the EGFR gene. And I will have to say that, as far as the details of this report, generally when we see an actionable mutation, we will think about what targeted therapy would be a good treatment option for our patients. Variant allele fractions so far in lung cancer don't play a huge role in influencing our treatment decisions because, while it's reported, it's not information we necessarily use because if a mutation it's there, and we use that information to guide our treatment decisions. And then moving on to the other sections of the report, there is, for this Tempest report, a section called tumor normal matched analysis. We don't often use this in lung cancer because in this section, it's really comparing the tumor molecular profile to a normal tissue like blood, just to look for any hereditary mutations or genes that can be passed down from parents to children. But I mention it here because it does show up on the report. As far as the pertinent negatives go, that's another section that shows up on the report. And what this is reporting on any clinically relevant genes in lung cancer that are negative. Remember that pie chart that I had showed earlier, where there's a whole number of molecular alterations that can be found in lung cancer and have associated therapies. Well, it's also important to exclude those alterations to make sure that they're not present as well. The other section of the report is immunotherapy markers. Here, what the report shows is a tumor mutation burden, which is the number of mutations in the DNA of tumor cells, as well as microsatellite instability status, which indicates the ability of it indicates the ability to repair mistakes in DNA. While there's a lot of information on the report, I think that, as far as the details of it, I think it's important to review it with your medical oncologist to really determine what information is important and useful in determining treatment options. Because for medical oncologists who are treating lung cancers, we're primarily focused on the genomic variants part of this report. The immunotherapy markers like tumor mutation burden and microinstability status don't have as big of a role in us deciding treatment options as the genomic variants do. The other part of the report that I think is also important to highlight is this FDA-approved therapy section. And what you can see in this report is that this is a list of the approved therapies based on this patient's molecular profile. So here it's listing a number of EGFR targeted agents, and here on the left side, it's describing the mechanism or how that drug works. But what I want to caution is that, even though here on the report, it lists a number of therapies like afatinib, dacotinib, osimertinib as FDA-approved therapies for the EGFR mutation, this is only listed in alphabetical order. It is not meant to be listed in any kind of preferred order of treatment. So, in practice, we're using a lot of osimertinib for our patients with the common EGFR mutation. So again, very important to go over this information with your medical oncologist to determine which therapy is appropriate. Just because they're listed here does not mean that every treatment is equal in efficacy and side effects. So that's why it's important to, review the report with your doctor. And then even for the FDA-approved therapies, there's different levels and categories of evidence here based on the National Cancer Guidelines, as well as the MSK OncoKB here that's listed on the report. But basically what it's, what that level of evidence or category of evidence is telling us is, what is the FDA approved status of the drug, and what is the level of evidence that we have for that specific therapy. So generally level 1, category 1 drugs have the highest level of data for those drugs, whereas, for level or category 4 drugs have the least amount of data or evidence for that drug. And that goes for the levels for the OncoKB consensus, as well as the NCCN categories for the different drugs that are available. And then at the bottom of this report is this clinical trial section. And that can be helpful because this can provide information about what clinical trials are available based on the molecular alteration that is found. In this report, this is reporting the different clinical trials that are available locally. So since I practice in San Diego, that's why we're seeing here the clinical trials that are located in the San Diego area. And that can be helpful to discuss with your medical oncologist when, when you're thinking about clinical trials, either in, upfront in the first, when you're starting therapy, or thinking about clinical trials when some of the initial therapies stop working. The other section that I wanted to go over that shows up in the molecular testing report is a section that will highlight mutations that fall under the category of variants of unknown significance. Basically, when we're performing sequencing of the tumor, we can pick up a lot of other molecular alterations, but there's a lot of alterations that we still don't know what the role is in cancer development and growth. And so for those alterations, they get listed alterations, they get listed under the category of variants of unknown significance. So while they're there, we don't quite have enough information about them at this point to know what their role or significance is. And as Dr. Marks had mentioned, as far as molecular testing in lung cancer goes, there's DNA and RNA sequencing. And for a lot of medical oncologists, we're now doing combination RNA and DNA sequencing because RNA sequencing can identify some gene fusions that DNA sequencing may miss. And so in the earlier report, I had reviewed a report for DNA sequencing, but if your medical provider orders an RNA sequencing, you might see that as part of the report. So just to make note that, as far as how we're identifying all of these molecular markers, there's a lot of different tests right now that are available to really try to find molecular targets in lung cancer. So that was an example of a molecular testing report in someone who got a tissue biopsy. But what about someone who we did a liquid or blood biopsy on, meaning we did molecular testing on the blood? So here is an example of a patient who had molecular testing done in the blood. Similar to the tumor biopsy report, this is a summary page where it's highlighting the molecular alterations that were found in the blood. So in this patient had an ALK fusion. And just like the other tissue report, this report also lists the FDA approved therapies for this specific ALK fusion. The report also mentions other biomarkers that are detected in the blood. In this patient, it's TP53. Just to make note, in the blood, the report will also sometimes mention copy number, which is just the number of copies of a specific alteration as a percentage of the total cell-free DNA in the blood. Again, for that, that's really primarily, it doesn't necessarily influence our treatment decisions, but again, important to talk about with your medical oncologist. But right now, we're not using the copy number to really decide our treatment recommendations. The other part of the molecular testing report on blood that was also seen in the tissue report was this tumor mutation burden here that's listed, as well as the microsatellite instability status. Here you can see that in the blood biopsy, they mention a tumor fraction, which just, which is basically the amount of circulating tumor DNA as a percentage of the total DNA in the blood. And very similar to the tissue biopsy report here in the bottom of the page, we can see here that In the blood, we find other alterations, but we may not know what their clinical significance is. So here on the bottom of the report, they're mentioning a list of those mutations that fall under that category. The other thing that Dr. Marks mentioned about liquid biopsies or blood biopsies is that they're non-invasive, and so because it's taken from the blood, we can get blood biopsies done at different time points. And so on the report, what you might see here is that if you've gotten a blood biopsy before and then got another one later on, sometimes the report will mention the change in the amount of copy number of that specific alteration over time. So you can see here in this report that between October 2023 and October 24, you can see how some of the levels of that copy number can change. But and that shows up here for this specific report as a tumor response map. The other thing that will be helpful as far as getting another blood biopsy is identifying any new alterations that can show up as a tumor changes with initial treatment, so that can show up on the report as well. So in summary, molecular testing really has helped patients with lung cancer. It's really helped us personalize cancer treatment with patients with lung cancer. It can be done both on tumor biopsies or blood biopsies. As the technology for molecular testing advances, we're now, doing both DNA and RNA sequencing on tissue biopsy. And again, it's very important to review that information from your molecular testing report with your doctor to determine the most appropriate cancer treatment plan. It's a lot of information in your molecular testing report, but it's certainly very helpful information. And I hope that this really empowers you to bring that information to your doctor so you can make a shared decision making about the best treatment plan for you. All right, thank you.
Thank you so much, Dr. Yoon. And if I may have all the speakers join us back with the cameras and audio signal. Thank you, everybody. So I think we have about 9, maybe 10 minutes for the panel discussion. And I'll start with my first question. So you know how we all talked about liquid biopsy, tissue biopsy. How do we approach the discordant results between liquid biopsy and tissue biopsy? Like, say, you see something in the tissue, did not see it in the liquid, or other way around, you see something in the liquid, and then you don't see it in the tissue. What does that mean, and how do you approach that situation? Whoever wants to start, and then we'll go from there.
Yeah, I can start with answering that question. As far as some of the discordance between the tissue and the liquid biopsy, sometimes what we can see is that if we find something on the tumor biopsy, but we don't find it in the blood, it can just mean that there wasn't enough of the tumor DNA to be shed into the blood. So that's why there can be the discordance there. Now, as far as, the other way around, why we may not find, while we may find something in the blood, but not find it in the tissue, it can just be that, as far as the biopsy goes, we picked a spot where, we picked a spot that maybe missed or didn't have, that tumor or tumor spot just didn't have that mutation, just because the tumor can show, heterogeneity, can, have differences in its molecular profile based on sometimes where we biopsy. And so that's where we can see the discordance there. I think that as far as, the specific mutations that I see either on the tissue or in the blood, if there's a mutation that's specific enough for lung cancer, and, say this is a patient with metastatic or advanced disease, I would use that information to go ahead and start treatment, because if that mutation is there, I would be, I think that would be enough information for me to utilize a targeted therapy.
Thank you. And I think the next question I'll maybe ask to Dr. Marks. Can you guide us through what is your molecular testing that you order depends on the stage of the patient? And I know it's a controversial topic, and so I think I'll have you start, and then we can maybe, if other people differ, other opinions differ from yours, then we can have other people chime in. Go ahead.
Sure. So, I mean, I think, yes, of course, you're going to see clinicians, including myself, doing molecular testing on patients in the advanced setting, especially because that's a clear indication to definitely do tissue comprehensive profiling on those patients. But as we continue to move treatment up earlier and earlier, especially with EGFR trials that have read out, we're testing for ALK and EGFR somewhat upfront sometimes in order to determine whether or not we're going to do a more neoadjuvant approach in the front line setting for an early stage where the cancer's still resectable versus going to surgery immediately and then having a treatment option after surgery where we have FDA-approved agents in that line of setting, specifically EGFR and ALK. So that is, of course, a controversial topic, and I'm sure you'll see people doing a number of things. I think the earliest, at least for me personally, knowing that information early is helpful and informative. And so I would like to get it personally as soon as possible in order to direct my treatment.
And I think it's, I agree with you. I think there is a minimum requirement and there is something that we all like to do. And I think at the minimum, if you do not have stage 4 lung cancer, at the minimum, you need to be tested for the EGFR and ALK. Because we do have FDA-approved treatment. For stage 4, it's your molecular testing needs to be definitely more expansive because we have more treatment options for those patients. And I think kind of going into kind of question to Dr. Sen, where we are right now is basically thanks to people like you who developed and study cancer for a living. We are just kind of on the tail end trying to use the drug. What can the patients do to help with translational research? Because our audience is patients, right? So what can they do to help you and eventually help themselves, because this research hopefully will translate with to approved drugs?
Thank you so much. And I would say it's a partnership. Yes, we try to do a lot of the discoveries in the lab, but it's truly a partnership with clinicians like you who take it to the clinic. So I think what the patients can do, and I see it more and more through these events that I go to, that the patients are really trying to understand the biology of the disease. And I think that makes conversations really easy. Another thing that I would urge everyone who's listening is that we need research material from the patients, right? We disease from the patients. So, if there is a consent protocol that you can, if there is a protocol at the institution that you're getting treated where they're asking for your tissue or your blood, your consent to participate in research, I would really request the patients to do that. If the clinicians are approaching you, understand so much more from the tissue or the blood itself with cell lines or animal models. So for the patients, I think participating in research through their tissue or blood is very important. Another thing is, talking to researchers about the new discoveries. And I think this is where these conversations are so easy and important, is when we break down what we do to what that contributes to. So research is expensive with the funding crunches that we are seeing, and with everything that the research needs. I think advocacy is really, really important, and that I have seen patients with lung cancer do that so much. But really advocating for funding for research is very important because that's how we will be able to progress in the clinic. So participate protocols in your institutions, learning more about the biomarker and advocating for patients with lung cancer.
Thank you so much, Dr. Sen. One more question I would have to ask you. So it's a two-part question. I'll start with the first part. If you are seeing a patient with stage 4 lung cancer, at which point should they get the molecular testing? And is there a special circumstances that will make you want to start treatment without knowing the molecular testing results? And is it wrong to start treatment without knowing the molecular testing results? So if maybe this time let's do Dr. Marks first, and then I'll do Dr. Yoon afterwards.
Yeah, so I mean, I think if a patient is symptomatic, where there's a large burden of disease, you don't have time to wait. I think you should be still getting that information in the background. I would certainly get a ctDNA immediately to try to inform that decision as quickly as possible. But if a patient is having a lot of symptoms from their actual disease, I would err on the side of giving chemotherapy alone in the metastatic setting, at least for the first line, or at to buy myself some time to wait for that test, but also treat the patient while saving that immunotherapy at the risk that if I give them in the wrong order, or if I identify a molecular alteration after I give immunotherapy, they're more likely to have toxicities associated with targeted therapy if I'm switching that quickly. So that's one of the few cases that I'll treat early, but love to hear other people's thoughts as well.
Yeah, I agree with Dr. Marks. I would try to get the molecular testing as soon as possible. So if there's a biopsy, I would send it for tissue molecular testing and would try to get the blood biopsy, liquid biopsy as well. And for a patient who's symptomatic or if their tumors, growing really quickly, but we don't have the molecular testing results, we can always start with chemotherapy. But certainly, with a patient who's, maybe female, never smoker, we really need to be on high alert for those patients who will likely have a molecular alteration. So that's why sending the molecular testing early on so we can get that, process going is going to be very important.
Thank you. And I think I'm probably going to start the Q&A session. There are several very interesting and important questions, and if we have time left over, I still have one more question I can ask the panel. So the first question that I think it's very important, is most doctors might be testing for biomarkers in adenocarcinoma, but not squamous. Why is that? And is this changing? And how do you approach patients with squamous cell carcinoma when deciding on doing or not doing molecular testing?
Yeah, so as far as the molecular, the targetable molecular alterations go, they do more commonly occur in the patients with adenocarcinoma. But, as far as my practice goes, even those patients with squamous cell carcinomas, there's still a very, while the fraction of patients with targetable alterations are much smaller, I would still do molecular testing in all patients who have non-small cell lung cancer.
Yeah, I mean, I agree. I think especially in squamous, if we don't look, we can identify a target. So, and those patients with squamous also it could arguably have, especially a BRAF or a MET exon 14 skipping. Those are known to be in smokers and in squamous histology, among others, while less common compared to adenocarcinoma. So I think removing that bias is also very important. Removing the bias about a smoking history, because I've certainly been surprised myself when I'm meeting a patient, getting a history, and while that can help, a little bit in predicting whether or not I suspect someone to have a target, I've been very surprised to find an EGFR in a patient I didn't expect. So
I agree. And I think especially like if any of you on the call have squamous cell carcinoma and you have a minimal history of smoking, there is a pretty reasonable chance that you might be having an ALK fusion or EGFR mutation. By the way, the first ever patient with ALK fusion that was described by Dr. Soda was actually a squamous cell carcinoma patient. So I agree with everybody. And another thing we haven't touched upon, but I think it's important. Yes, we might not be having a lot of approved drugs for molecular alterations in squamous cell carcinoma, but it also opens an opportunity for a clinical trial, right? So if you find a mutation that might not have an alteration, but there's a clinical trial available for that mutation in your immediate area, I think that would be beneficial as well. So I think there are a couple of other questions which I would want to see. I think one is interesting. Do you see next generation sequencing being aligned to federal standards at some point? And anybody wants to take that? I can start. So number one, there is no federal standards, right? So there is no federal standards of what we do in testing. We do have federal standards, which is FDA when we approve the drugs, but nobody regulates which patient needs to be tested. We follow guidelines, and we have national societies like ASCO, and in non-solid tumors, it's ASH, which provides guidelines, which is an expert opinion statements on what patients needs to be tested. And then number two, the one that's couple of us, I think Dr. Yoon and Dr. Marks, I think showed something that's called NCCN guidelines. So NCCN guidelines is another guideline committee. It's again a consensus guidelines. They are very difficult to read, to be honest with you, because it has a different purpose for us. But if you go on NCCN guidelines, there's actually patient version that is a little bit more distilled down and easier to read for you. So I would highly recommend for us to do that. I think another question I have, and then maybe I will have either Dr. Sen or Dr. Marks answer. So Dr. Yoon, you mentioned percent of tumor fraction when you look at the cell-free DNA. And you mentioned that really at this point, we really don't make a decision if it's like 7% or 14%. We do make a decision if it's a 50% because that means something to us. And I'll have maybe Dr. Sen and Dr. Marks explain what that means. But, is there a minimum number that we need in ctDNA or liquid biopsy to believe that a mutation is truly there?
Yeah, I don't think that there's like a hard set percentage that we necessarily need. But I think the higher the number, then, then it tells us that there's more of the tumor DNA in the blood. And so based on that report, then we're seeing more of the, the tumor DNA information. And I think that, if we don't see something in the blood and the tumor fraction is low, right? Like, are we really convinced that there's enough tumor DNA there to say that it's negative? So I think that's where it can, that tumor fraction is kind of informative for oncologists.
And can somebody take the question of what does that mean if your variant allele fraction is like 49%, 50%? What does that mean for the patient and what do we need to do?
do you want to take this, Dr. Sen?
Sure, I can talk about, what does that mean? So tumor fraction, we mean the percentage of circulating tumor DNA in that blood sample. So it is a biomarker, as my colleagues have mentioned. So it's really analyzing the amount of ctDNA, and it's a ratio of the circulating tumor DNA to the cell-free DNA that we find. And it can depend upon a lot of factors, which could be anything between, the allele frequency, fragment length, aneuploidy. So it's a lot of things that is taken into consideration into making these decisions. But in a broader term, it's really the percentage of circulating tumor DNA. That's what we call as a tumor fraction. I don't know if that answers the question.
Yeah, I guess the last part is if it's above 50%, I think that would validate or warrant more testing. Specifically, that would be very suggestive of a hereditary or genetic alteration. So that requires a whole nother set of testing on a different platform, but especially to inform genetic counseling decisions. I think in that case that would be important to go in that direction and make sure you get those additional tests.
And Dr. Besbro, your previous
Go ahead, Dr. Sen.
Yeah, your previous question about liquid biopsy versus tissue. I would also like to mention, the disease matters in small cell. When you do tissue-based, you look at mutations, right? Which is not, right now, we don't have targetable alterations in small cell lung cancer. And I think liquid biopsy in that area can really help us in understanding the progression of the disease and seeing when the disease recurrence. So I think for small cell liquid biopsy, and since the tumor sheds so much CTC into the blood, the circulating tumor cells into the blood, I think liquid biopsy and the disease a lot more. And I think in that case, not just the DNA information, I think we have to go a little deeper into the RNA and the protein information. And that's where we are going next, right? So going to the next level based on the disease that you have. How can we tailor it to the disease?
Yeah, and I think what Dr. Mark said, I completely agree. So if you look at your liquid report, the liquid biopsy, and you see the VAF next to like 45, 46, 50, 52, definitely talk to your oncologist, because that means that you might have a germline DNA abnormalities, which will not really affect what we do for you, but it might affect a desire of your first degree family members to be tested as well, right? So we see Lynch syndrome. There are some inherited EGFR mutations that they're very rare, but we see it once in a while. Looking at the Q&A session, just for our audience, I see a lot of questions which are specific to the mutation. And then later in the day, we're going to have a panel discussion, not panel discussion, but presentations with Q&A for a specific mutation. So I'm going to leave those questions for those parts. The one question that keeps coming up, and I'm glad that we have Dr. Sen in there, because that's what she does in her lab, is question for small cell transformation from the non-small cell lung cancer. Should the molecular testing be done after histological testing? I've been told by my oncologist, since there are no targeted therapies available in Poland, this question is originating from Poland, there is no sense to do that. And it's a pretty controversial question, to be honest with you. And I'll start with Dr. Sen, and then maybe if the other panelists can chime in as well.
Yeah, just to orient everyone, small cell transformation is when either an EGFR, ALK, and now even we are seeing with KRAS, when these patients get treated with the tyrosine kinase inhibitor for that oncogene, so for EGFR, it would be osimertinib. When they get treated and they develop resistance, they actually transform to small cell lung cancer. And it's right, because when the disease comes back as small cell lung cancer, most often, I think in almost all cases, the EGFR mutation will still be retained, right? We will still have the EGFR mutation, along with, in most cases, we see RB1 and P53 alterations as well. So yes, once you have histological transformation, I think molecular testing, we don't have ways to treat it, because they're treated like small cell lung cancer at that point, right? They're treated with chemotherapy and other things which would be a treatment for small cell lung cancer. So in that case, apart from the mutation, which is EGFR or ALK, which is still retained, molecular testing, still we don't have anything to do with that information because we don't have targeted therapies that can go for that cancer type. So I think till now, molecular testing for that percentage of patients could be not actionable at this point. And I see another question, which is to do with the same thing. For small cell transformation, should TKI be kept going along with chemo? So, when we did the testing in our clinical samples, we saw EGFR mutation was still retained. So in my opinion, you would still have to keep the pressure on the EGFR to get the best. So I would say, if it's an EGFR mutant patient, then having osimertinib in the mix would actually help because that helps control the EGFR part. And then you come in with a combination, which is to treat the small cell lung cancer tumor.
Yeah,
Yeah.
thank you.
I completely agree, is the short answer.
Yeah, good. There are a couple of more questions that I think it's important. They're coming regarding liquid biopsy. And the first question, which I think it's important to reiterate again, even though Dr. Marks, you did mention it in your talk. So is obtaining tissue from the resected specimen, in this case, or any kind of metastatic specimen, provides more in-depth testing, which is not available on a liquid biopsy. If you can just highlight again the difference, what does liquid misses and what does it gains, and what is the tissue gains and what it can be missing? Because I think it's very important.
Absolutely. Yeah, no, that's a great question. So with comprehensive testing on tissue, ideally we're getting a next generation sequencing with looking at DNA sequencing as well as RNA sequencing. So this is identifying smaller mutations when we're doing them complementary together. And so when you get that report back, you're not just getting, information about the one single gene. You're getting often whole exomic information and whole transcriptomic information to really identify what's going on with the tumor as a whole. Some platforms have PD-L1 incorporated into that, but all of these individual things help to inform treatment decisions. And so I guess the other thing is liquid biopsy specifically, most platforms, if not all that I'm aware of, unless they're not necessarily approved yet, are only using DNA next generation sequencing. And so not only is the shedding of the tumor, you have to have a high enough tumor burden to detect an alteration, but it's only using one of those modalities, that being often DNA next generation sequencing. So you could miss a small indel or like the MET exon 14 skipping mutation on a liquid biopsy that you might be able to detect on tissue. And so they need to be used together and with caution, with all of the potential pros and cons about missing such alterations. Last thing I'll say is, for example, if you're doing a biopsy and you get a specimen that shows a KRAS mutation, it's becoming more important. Well, what KRAS mutation is it? Is it KRAS G12D, or is it V, or is it C? We only have targeted therapies for certain mutations right now, but that we also have clinical trials in those spaces too. But then STK11 and KEAP1 are co-mutations that may inform further treatment decisions. And so the comprehensive testing can help to guide your clinician, and you might not get that whole profile with just doing the liquid testing alone.
Yeah, and building what you just said, there's a question just popped up, which I think is perfectly aligned with your answers. What does having P53 mutation adds to the treatment or prognosis, and just kind of explaining why you would need comprehensive molecular testing. Anybody wants to take that?
Yeah, I mean, I think I'll start. That's a complex question because it's controversial in certain subtypes. I mean, traditionally, and Dr. Sen could speak to this probably a little bit more, EGFR and P53 portend maybe a worse prognosis. If there's an RB1 mutation, that might be more likely that that patient's going to transform it or has a risk of transforming into small cell lung cancer. So it's also important to get biopsies if patients are progressing on an EGFR TKI or targeted therapy alone. But I guess with MET exon 14, it's not necessarily prognostic in that case, and still more research is to be done there.
Yeah, I'll just add, and I completely agree with Dr. Marks, is that with an EGFR mutation, if there is a P53 loss, and if there is an RB1 loss, sometimes RB1 cannot be detected in mutation, and we have to do protein testing for that. But if there is a P53 mutation, I think that patient should be monitored, and if and when they develop resistance, there's a highly likelihood that they would develop resistance through histological transformation. And I would say in that case, we haven't reached the level in liquid biopsy for that detection, and tissue remains our gold standard because we have to look at neuroendocrine markers and other markers in our tissue. And at that stage, I think tissue is what we should be testing. We are not there yet with the liquid biopsy part.
Thank you so much. The next question I'd like to ask Dr. Yoon, that's kind of going through your molecular report, and I think our listeners probably have a better understanding of what they're looking at. I think this question is very, very timely and important. If a hospital that you are at is using a single gene testing, and not NGS, but a single gene testing, and does not find any mutations, can they safely assume that the person has no mutations and make a treatment decision based on it? And if so, isn't this risky? So basically, can you highlight the difference between a single gene testing and next generation sequencing, and what does next generation sequencing brings, and what the pitfalls we have in a single gene testing?
Yeah, so for single gene testing, it's in the name. So we're only looking at a single gene. Whereas, for the next generation sequencing, we're looking at, the entire sequence to look for, a bunch of other alterations. So just because, a single gene test was performed and that's negative, we can't assume that there's no other alterations just because we haven't tested for them. So in that situation, I wouldn't just rely on a single gene test result, if that's negative to provide, my treatment recommendations. I would like to have, information about other genes and do a more comprehensive sequencing to look for other alterations. But, as we talked about before, if there's a time where like a patient is very symptomatic, where we need to start chemotherapy, that might be a situation where we might start some upfront treatment. But, ultimately would want that information with whole gene, next generation sequencing to really do a more comprehensive search to look for other alterations that can be acted upon.
Thank you. And especially like we published in the setting of EGFR exon 20 insertions, that single gene testing for the EGFR misses the EGFR exon 20 insertions in a clinically meaningful number of patients that we missed. And I think the way I kind of like to use non-medical explanations when I talk to the patients, like for example, if you are entering your closet and you have a mind that you want to get a red t-shirt, you're going to completely miss the green one because you were looking for the red t-shirt. So that's why I think it's important for us to perform next generation sequencing. I don't know if Dr. Sen, you have anything else to add from your basic science standpoint?
No, I think they really answered the question well. I don't think I have anything to add to that.
Yeah. And another question, I think we have about 2 minutes. This is a question that puzzled me initially as my first like looking at now. I think I kind of I know what you say. Does the age of the patient influence the accuracy of the liquid biopsy? For example, is the liquid biopsy more accurate in the younger patient? And to build on that question, what makes the liquid biopsy more accurate?
I can just talk a little bit about the mutations. So, as the patient's age, we know from several studies, tumor suppressor genes, the mutations in those can happen with age. So P53 loss and others can happen with age. So when we are doing liquid biopsy on an aged patient, we may have, through the process of aging, they may have gathered certain mutations in the tumor suppressor genes. So whether they're true oncogenic or whether it's a process of aging, I think that's where some of the false positives can come in. So, I would like to say that age does add complexity when we try to look at the liquid biopsy results.
Yeah, I mean, I think you might be detecting smaller mutations that develop with time, like chip alterations that don't necessarily, or they're not actually influential in terms of your treatment at that time, but certainly contribute to the complexity, as you said.
Yeah, and I think what we know, what makes liquid biopsy more, not valuable, but more reliable is the disease burden. That's what Dr. Mark said. And I think, again, non-medical analogy, right? So if I completely owe that analogy to Dr. Molly Lee from Hong Kong. So if you are fishing and you put the rod in and you didn't get anything, that doesn't mean that there is no fish in the sea. So it just means that you're either a bad fisherman, meaning using a wrong test, or there is not enough fish in the sea to get it on your rod. So that's how you think about the biopsy. Negative liquid biopsy does not mean you don't have fish in the sea. All right, I think we are at time. First of all, I'd like to thank our panelists for spending their Saturday work-free day with us to give education to our patients. And thank you, everybody. That was wonderful. And then we'll move to our second session. Thank you very much.
Thank you, everyone.
Thank you.
Okay, so I assume we're ready to go for the second session. So give me just 1 second. So our first speaker in the second session is Dr. Melina Marmorelis, who is an assistant professor at the University of Pennsylvania and the medical director of the Penn Medicine mesothelioma program. Even though she's a director of the mesothelioma program, she's a force in lung cancer in general, and she specializes in thoracic medical oncology. She does a lot of phase 1 clinical trials and molecular testing clinical trials. So welcome, Dr. Marmorelis.
Thanks so much for having me. I'm going to pull up my slides. Great. So I have the honor today of speaking to you about what is targeted therapy. So we're going to go through a few different parts about this. So first, I just want to take a step back and understand what are the different tools that we have when we're thinking about treating lung cancer. So we do have some traditional medicines that we think of, or medicine, quote unquote. First being surgery. Can you cut it out? Is it something that we can cut out, and is cutting it out going to help the overall state of the disease? Second is chemotherapy. That's an older anti-cancer medicine. It's a chemical that goes in and interrupts the cells from growing. It targets a lot of different things, can be very effective, but is one of the older anti-cancer medicines that we have. And then radiation, which we're going to hear a little bit more about later in this session, where we use an external beam to target things. Again, asking a similar question that we ask when we're thinking about surgery: can we target something, and does that help the overall disease state, or does it help with a symptom that a patient is experiencing? And then on the other side of this, we have precision medicine, and that's what we're going to focus on today. I'm going to talk about immunotherapy first because that's not the part of targeted therapy I'm going to talk about in general. But it's also a type of targeted therapy. It targets the immune system and stimulates a patient's own immune system to fight the cancer. So it's unlike chemotherapy, it's not trying to kill cells, it's actually just mobilizing the troops that a patient already has and hoping that that will help to provide some anti-cancer therapy. And what we'll spend the most time talking about is targeted therapy, where we're taking advantage of certain aspects of a patient's tumor to slow tumor growth and kill cancer cells. And these come in actually a variety of different types. So they can work in a few different ways. They can stop cancer cells from growing. They can change proteins in a cancer cell so that the cancer cell dies. Like immunotherapy, they can trigger the immune system to actually kill cancer cells, or they could actually just be finding the correct cell, but carrying with them the thing that will kill the cancer cells, like something like a chemical, a chemotherapy, or a radiation. And we use this in conjunction a lot of the time with some of those other therapies that I mentioned. And lung cancer is actually really the poster child for targeted therapy, which is very exciting for us. On the left, I'm showing the rates of death from lung cancer. And you can see it's actually the purple with the arrow next to it. And the survival rates are actually really improving. And part of that is due to being able to find targets in tumors and to be able to target them. So this other graph shows a variety of the different targets that we have and the things that we look for. You guys talked a little bit about molecular testing in the last session, and this is the reason why we look for those targets, is because we do have targeted therapies that can target them. All right, so let me go ahead and here's the progress that we've made. Back in the early 2011, 12, 13, 14, we had a few targeted therapies: crizotinib, erlotinib, afatinib, ceritinib, and we were able to target a few targets. But really, things have exploded in the last 10 years. So we have too many to name, and if I included 2024, we would also see additional agents that were approved in 2024. So this is by far and away the most targeted therapies seen in any cancer. And we're really proud of that. We're really proud of that progress and proud that we're able to offer that to a lot of our patients. There are different types of targeted therapies. So there are monoclonal antibodies. Those are usually given through an IV in the arm in an office, and they target something on the outside of a cancer cell. So they are attaching to it. Maybe they have something attached to them that can actually get into the cell, or maybe they're just bringing different cells together, but they're working on the outside of a cancer cell. And then there are small molecule inhibitors, and those are actually pills that a patient takes at home, and those are getting into the inside of the cell. Sometimes they're interrupting processes of replication. Sometimes they're interrupting other processes within the cell. So who might benefit from targeted therapy? The first key to that is molecular testing. So here again, we have the pie of all of the different things that we look for, and that many of which we can target. So now all patients with non-small cell lung cancer should get molecular testing. These are more commonly found in patients with adenocarcinoma and with a lighter no smoking history. However, that paradigm has really shifted recently as we have discovered how to target things like KRAS G12C, which are more common in patients that have a smoking history. So really, everyone should get molecular testing. It's important to understand and to know what the molecular characteristics of your cancer are. And how do we test for these targets? You guys talked a little bit about this in the last session, but you can either get a piece of the tissue, a standard biopsy. That sometimes takes time, it's invasive, biopsies have risks, or you can also get a blood sample and look for pieces of the tumor DNA in that blood sample, or a liquid biopsy. That often comes back quicker, but again, not everybody has pieces of the tumor DNA in their blood. So often we like to use both of these so we have as many possible chances to detect something. Targeted therapies are not a free lunch. They unfortunately do have a number of side effects that are real and can affect patients' quality of life. Probably the most common is a skin reaction. They have a decently high rate of causing rashes. There are some that can affect mood or cognition, some that can affect your sleep. Many of them do upset the gastrointestinal tract, so that can manifest as either constipation or diarrhea or just frequent bowel movements. It can cause fatigue, having lower energy. And some can affect your blood pressure, so causing a higher blood pressure, or really other cardiac issues like low heart rate, those types of things. So those are all things to consider when we're thinking about the different targeted therapies, and also things that we monitor when we're talking to you about what you're experiencing on a targeted therapy. But there are some actually really unique advantages of targeted therapy that are special to this class of drug. One is, I mentioned, most of them are actually oral medications. Most of them are pills that you take at home. And actually, they're pretty well tolerated, especially as we get later into the development of these targeted therapies. We're able to target with more specificity, and we're not targeting, we're not having more off-target toxicity, which basically means we're not hitting the things that we don't want to hit, the normal cells. And one of the real benefits, especially of the small molecules, in particular the pills that are targeted, is that they're so small that they can sneak their way into the brain and sometimes affect disease that's in the brain. So that's something that we really look for and is a huge advantage of targeted therapies. We're going to hear next about resistance pathways, but targeted therapies can also be used to target resistance pathways if we can figure out why a patient has become resistant to a certain target, to a certain therapy. So let's talk about kind of what's happening now and what's coming next in targeted therapies. So I wanted to mention a few areas that have really had a lot of development in the last few years. So one is a rare mutation, a rare EGFR mutation. So this is EGFR exon 20 insertions, previously very difficult to target. And initially, a lot of the pills that they tried didn't work very well. And in the last few years, we did have an infused targeted therapy, amivantamab, that was approved for this target, which was very exciting. And now on the new frontier, we have a number of actually pills that look like they might be affecting the disease. These are waterfall plots, which basically show if it's going down, it means the tumor is shrinking. If it's going up from the 0 line, it means the tumor is growing. And what you can see here is that these look like waterfalls. They're going down, meaning that these drugs are overarchingly decreasing tumor volume, which is very exciting in this previously very difficult to target group. Another tricky target has been HER2. So HER2 has been a target in breast cancer for a long time and is probably the original targeted therapy if we think about targeted therapies. But in lung cancer, it's been really, really tricky to target. They tried some of the old targeted therapies that are used in breast cancer, and they didn't really work very well. So as we got to newer targeted therapies where they're using both an antibody as well as the antibody is now linked to a chemotherapy. So this is an example of an infusional treatment given through an IV, an antibody that attaches to the outside of a cell, and it's carrying with it a chemotherapy that's going to kill the cancer cell. And here you can see again, we have a waterfall plot where most things are going down, showing that the tumor is shrinking. And these are patients with HER2 mutations. So again, molecular testing identified this mutation, and we were able to target it with this targeted therapy. And then KRAS G12C, which really was thought to be untargetable in the past due to its conformation. It was hard to design a drug that actually got into the right places of this protein. But they were able to do it. The first approved in 2022 adagrasib, or 2021 sotorasib, and then 2022 adagrasib. And this was very exciting. This felt like one of the first nuts that was cracked in the targeted therapy space for these hard to target alterations. And I think really opened up the door to a lot of other targeted therapies. KRAS G12C mutations can be found in any type of non-small cell lung cancer, adenocarcinoma, or squamous cell. So that's a really exciting part of this target. And I think the other thing that's interesting about this one is that chemo and immunotherapy can still be really effective, just as in some of the other targets, but even more so here. So we have a lot. It's nice to have additional options here. So I just wanted to talk about a few questions to ask when you're thinking about a targeted therapy, or you're talking with your physician or your team about the targeted therapies. So, what type of targeted drug will I get? Is it going to be an infusion, or will it be a pill that I take at home? How often will I get the treatment? Am I coming into the office? Am I taking it daily at home? Do I need to go somewhere to get my treatment? How long will my treatment last? Some of the infusions vary in time, so different burdens of time to get those. Will my insurance pay for my treatment? How much will I have to pay? So it differs whether you get it in an infusion area or whether you're taking a pill at home. Side effects are obviously very important to talk about. How can they be managed and what can I do about them? A lot of the targeted therapies have more side effects within the first 6 weeks. And so it's really important to have close communication with your care team during those times. Are there any special things I need to do to protect myself and others while I'm getting treatment? So certainly things like talking about contraception, and how the drug is metabolized, those types of things. And then when should I call the doctor or nurse? When should I be worried? When do I need to ask for help? And certainly, even if you have a molecular target, you have to think about all of the factors that go along with targeted therapies. So what's the side effect profile? What's the intensity of monitoring? How often am I coming in? Is it once a week? Is it every 3 weeks? What if I do this treatment? What are my options later down the road? How will this affect the other medical problems I have? My hypertension, my diabetes, those types of things. Is this something that within my insurance policy is feasible for me to do, or in the area that I live, is it very convenient to get an infusional therapy, or is it more convenient to be on an oral therapy? And then certainly there are tumor characteristics that your care team is thinking a lot about. Where is the disease? What are the different subtypes? And these all go into making the recommendation for treatment. There are a number of amazing biomarker-focused patient groups. These are some of them, but it's been an amazing thing to watch these global communities come together, especially with some of the rare mutations. They provide a lot of support to patients, as well as accelerating research in these areas where it is actually hard to study their rare cancers. So I would really encourage any of you who know your molecular profile, and if you have one of these mutations, to reach out to these groups and participate, because you can really contribute, and they can also, I think, really provide additional value. So just a few final thoughts. We have seen rapid approvals of targeted therapies in subsets of lung cancer. It's a very exciting space. But of course, we need to do molecular testing in order to find these molecular alterations. And there is still a really important role for chemotherapy and immunotherapy in lung cancer, even if some of these are present. So these are not the only types of treatment that we consider. Thanks so much for your time.
Thank you, Dr. Marmarelis. And we'll leave the questions to the Q&A session. Next, it's my pleasure to introduce Dr. Eric Singhi, who is an assistant professor and a thoracic medical oncologist at the University of Texas MD Anderson Cancer Center. He specializes in patient education. This is a perfect avenue, and young onset lung cancer, and he serves as a co-director of the Thoracic Medical Oncology rotation for MD Anderson Medical Oncology Fellowship Program. Welcome, Dr. Singhi.
Thank you so much for having me. Thank you to Cancer Grace for organizing this. I really appreciate it. Give me 1 second to share my slides here. Okay, and that looks okay for everyone?
Yes.
Perfect. Okay. Well, it's been a wonderful lineup so far. I'm honored to be here with such amazing co-faculty. So thank you for the opportunity. My name is Eric Singhi. I'm a thoracic medical oncologist at MD Anderson Cancer Center. And today I'm going to be talking about understanding resistance to targeted therapy in non-small cell lung cancer and what that means for patients. These are my disclosures. So this is a brief outline of my talk. We'll very quickly go through Targeted Therapy 101. We just heard an amazing talk about that concept. We'll talk about types of resistance, types of disease progression, what tests are essential at the time of disease progression, how might you be able to treat or manage progression, and then some key takeaways. So we just saw this slide, so I'll be very brief about it. But really, this is about anchoring what precision medicine targeted therapy means for patients with non-small cell lung cancer. We know a lot about traditional medicine, what's really been the historic staple for patients with lung cancer for many years: surgery, chemotherapy, that's that traditional cytotoxic chemotherapy, radiation therapy, but now targeted therapy, which is a type of treatment that specifically targets any cancer-specific genetic changes to slow the tumor growth and potentially kill those cancer cells. Of course, what's really important to figure out if targeted therapy may be an option for you in your case is biomarker testing. And so there's unfortunately a big gap in biomarker testing. So continue to advocate and continue to raise awareness about how important biomarker testing is in non-small cell lung cancer. It's really going to help identify tumor-specific changes, which we've talked a lot about, like these mutations, and it's going to determine if a targeted therapy is effective in your case. This is the reason why targeted therapy is so important. So as you can see, this is a list of all FDA-approved targeted therapies that are available for patients with non-small cell lung cancer based on the biomarker that we identify. And so these are some of the benefits of targeted therapy that were just nicely reviewed: more effective treatments for specific cancer mutations, fewer potential side effects compared to that traditional non-selective chemotherapy, improved quality of life for patients, a potential for longer survival rates, and that ability to personalize treatment, right? Which is really important. And so I like to call it the Ken of the trio, right? So we have chemotherapy, which tends to be more toxic, it's non-selective, immunotherapy, which we've been very excited about for a few decades now, and then targeted therapy, which really offers that opportunity to personalize treatment. Despite all of these benefits, there is a challenge, and the challenge that we're going to talk about today is resistance development, and that can really limit long-term effectiveness of the targeted therapies that we're prescribing for our patients in clinic. So the first concept to talk about today with regards to resistance is what, meaning what type of resistance might a patient experience with regards to being on targeted therapy. And so there is this concept of primary resistance. And so primary resistance is where you have resistance from the very start of targeted therapy. So if you look at this graph here on the right, you can see that this is tumor burden, meaning how much cancer is present at the start of being diagnosed with cancer. And you can see for this patient experience that unfortunately, despite starting treatment, the tumor burden, the amount of cancer continues to rise. And that's even at the start of targeted therapy. And so we call that primary resistance. Then there's a concept of acquired resistance. And so if you notice here, right at the beginning of diagnosis of cancer, there's a high tumor burden. There's a high level of cancer that's present in that patient. And what you see is that when they start targeted therapy, that level of cancer in the body starts to go down. And that's the response that we want. And so most patients will have this initial response, But then they'll still have what we call minimal residual disease, which means that there's still a very small amount of cancer that's present, maybe not even seen on the scans, and that continues to persist. And ultimately, at a certain time point, we start to see that there is that level of clinical detection, that amount of cancer that's present on the scans that we can see, and then it starts to rise, and we call that acquired resistance. So this is going to be really the main bulk of focus of our discussion today. So how do we know if targeted therapy is working in your case, is still working? Well, one is definitely symptoms. That's why we spend a lot of time with our patients at the bedside. We ask, how are you feeling? We encourage you to discuss any symptoms that you're experiencing while you're getting treated. And then we rely on our scans, our imaging test, and that's really our gold standard. Another thing that we think about when we're talking about clinical trials, we've talked about it a lot today. The reason why we found all of these targeted therapies is because there were patients and their loved ones many years ago who heard about a promising drug that was being studied on a clinical trial. And they courageously and very selflessly stepped up to say, I want to be a part of this, to see, one, is it going to help my case? And second, is it going to help patients who are facing a similar condition in the future? And we track different endpoints on clinical trials to see, is this drug working and is it still working? And so one of the clinical endpoints that we use often to see, is the targeted therapy still working, controlling the disease without it getting worse, is called progression-free survival, or PFS. And so I won't talk a lot about this today, but I really focus on patient education. And if you're interested in learning more about clinical trials, what they mean, how you might be able to understand them as a patient, I'd encourage you to check out this article that's in Future Oncology that I helped co-author, and it really explains endpoints in patient-friendly language. So we've talked a little bit about what types of progression might happen. Let's talk about how, and these are patterns of disease progression. So these are the three big categories that I generally explain to patients and their loved ones. The first is CNS progression, or progression in the brain. So what this means is that there's, unfortunately, despite being on targeted therapy, you're going to have growth of disease that's restricted to the brain, while the rest of the disease in the body is actually quite well controlled. If that's the case, and you can see in this figure here, you may have an opportunity to still continue your targeted therapy and then call in our colleagues in radiation oncology to radiate that concerning brain lesion and stay on the same treatment. So we'll hear more about this in the next session, but this is a little bit of a teaser. The next category or type of progression is called oligo progression. Oligo means few. And so what this really means is that cancer has spread to a few other areas despite you being on targeted therapy, but it's still mostly controlled in other parts of the body. And so this is an example of a patient who was on a targeted therapy, and you can see their disease, their cancer that's highlighted or circled in the red there. And this is before they start their therapy. 2 months into therapy, they have a really nice response. You can see, you really can't see any of that disease that's still present. And then unfortunately, with more time on targeted therapy, they develop acquired resistance, right? And they have this isolated spot that's right there depicted by the arrow. This is another opportunity to consider calling in our colleagues in radiation oncology to radiate it and potentially continue on the same targeted therapy. So stay tuned, we'll talk more about that. And then finally, the third type of pattern of disease progression that I explain to patients is called widespread progression. This is where, unfortunately, despite being on a targeted therapy that was working well, the cancer has learned how to get smart, and it's learned how to spread to many different areas, and it affects a larger part of the body. And so in this example here, this is looking at the lungs of a patient, and you can see that there's a lot of this patchiness on both sides. And so unfortunately, the disease is growing, and it's growing in a widespread manner. So we've talked a little bit about the what, we talked about the how, but now we got to figure out why. Why is a patient unfortunately developing disease progression despite being on a very selective, a very precise type of medicine, which we call targeted therapy? So this is the why, and it's a really complicated slide, so I'm going to break it down for you. One of the first mechanisms that we think about for resistance that develops on targeted therapy, right? So this is a patient who's on targeted therapy. This is showing you the signaling that's happening, is that these patients unfortunately will develop what we call on-target resistance. On-target resistance means that the cancer cells develop mutations in the same target that the therapy is aimed at. So I'll give you a specific example for our patients with EGFR positive disease. So you may have heard of some of the more classical or common EGFR mutations, like EGFR exon 19 deletion or EGFR L858R mutation. Well, unfortunately, in some patients who are taking targeted therapies such as osimertinib or Tagrisso, for example, they will develop an on-target resistance mechanism. And so an example of one can be the C797S EGFR mutation. It's actually the most common mutation that's acquired upon resistance to Tagrisso or osimertinib. Happens in about 6 to 18% of patients, unfortunately. But when we understand that on-target resistance mechanism, there may be options for therapy that we can aim and try to overcome that resistance. And so that's why it's so important to understand, is there an on-target resistance mechanism? Next, there's a potential for off-target resistance. And so this is what happens when resistance comes about from a change in other pathways that bypass the targeted therapy. And so another example, going back to our patient with EGFR positive disease, they're on their osimertinib, it's targeting that EGFR pathway, but the cancer gets smart and it learns to take a bypass route, a bypass pathway. And so these are some examples of other alterations in cancer that get upregulated and that allow the tumor to escape and sort of grow on that pathway. One of the more common ones in EGFR is called MET amplification. Happens in almost 16 to 18% of patients who are on Tagrisso or osimertinib. But once we learn that, there may be an opportunity like this box in green to really target it. So that's another example of why it's so important to understand. Finally, we have this other potential acquired mechanism of resistance that we call a lineage transformation. Dr. Sen and her lab do a fantastic job of really trying to understand why this is happening. And so what that means is you start off with non-small cell lung cancer, right? The most common type of lung cancer. And then the cells, the biology of the disease actually changes, where you have a switch in the cancer cells and it changes from one type of cancer, non-small cell lung cancer, to potentially small cell lung cancer, which is unfortunately a highly aggressive type of lung cancer. So this is a nice depiction of how that happens. Patient is exposed to the EGFR TKI targeted therapy drug like osimertinib. It's doing a really nice job trying to shut down those pathways that upregulate the growth of these cells. And then unfortunately, there happens to be some genetic changes, a loss in these genes such as RB1 or TP53, and it causes a change in the types of cells that are now causing trouble into small cell lung cancer. And then finally, there's one more sort of bucket that we think about for acquired resistance, and it's the ever unfortunate, really frustrating bucket, and that's called the unknown, where we just don't know yet. Dr. Sen and colleagues in the lab are doing a fantastic job trying to understand it, but these are generally the 4 potential mechanisms of acquired resistance to targeted therapy. Now, you've learned a little bit about the potential mechanisms, but how do we learn more about which one it might be in your case? And this is why post-progression testing is so important. So what do I mean by that? Well, this is a really nice diagram here that shows you, okay, unfortunately, a patient has resistance to their targeted therapy that we start off with. Their disease grows, and sometimes it can grow in that widespread manner, like I showed you, which we call systemic progression. What test can we do now? We heard a lot about some of the tests from Dr. Marks and others in session 1 about what we should do and how we should test for repeat molecular testing. And we can do it on a liquid biopsy, a blood test, but also a tissue biopsy. And so I would advocate and I would discuss with your oncology team, if your disease grows on the targeted therapy, we want to learn more. We need more testing. So we need more profiling. And we can do that in both of these different ways. And again, that information can help us figure out which resistance mechanism is happening in that case. And it's going to help guide decisions for your next line of treatment. When you get your results, there's really 3 potential options if a patient has widespread disease progression. One is clinical trials. Please ask about clinical trials with your team. If you have a local academic center nearby, ask for a second opinion, advocate for yourself. You won't know what the options are until you ask, right? So I think clinical trials should definitely be considered in this case. Next, you want to really try to target any specific mechanisms of resistance. I showed you on-target mechanisms of resistance, off-target mechanisms of resistance. Those can potentially still offer a targeted way to overcome that resistance. So really think about that as well. And then if you can't get onto clinical trial, we can't find a very specific mechanism of resistance to continue a different type of targeted therapy or add in a different type of targeted therapy, then we have standard of care options. For standard care options, chemotherapy is really our staple. It's our backbone. And we've talked a little bit about chemotherapy already. So when do you generally add chemotherapy if you have progression of disease on targeted therapy? Well, generally, what I would say is for patients who are having very rapid, very symptomatic disease burden and widespread progression, you want to consider adding chemo. Also, in cases of small cell transformation, that highly aggressive type of disease, unfortunately, our options right now are quite limited. And so we really need to rely on chemotherapy, which can work quickly and work well for patients. So chemotherapy is recommended if small cell transformation occurs. There is controversy, and we've already touched on it a bit, about whether you should continue your targeted therapy at the time of progression. And so I would discuss the pros and cons of that with your oncology team. To kind of round out this discussion and talk about what may be coming in the future, something that we're really excited about is antibody drug conjugates, or ADCs. And this is a new treatment approach, and we'll probably hear more about this later today. But what are ADCs? Well, ADCs are this new class of drugs, and they deliver chemotherapy in a really smart way. So they deliver it directly to the cancer cell. So this is a mean, angry cancer cell that I found. And this is an antibody drug conjugate. It's the blue, green, and then this red with a Death Star on it. But essentially, the antibody drug conjugate will have an antibody part that directly attaches to the cancer cell. And then it'll have this linker that's holding onto a very high chemotherapeutic cytotoxic payload. And it'll drop it into the cancer cell. And sometimes it can even go outside of just that specific cancer cell and kill some nearby cancer cells as well, which we call a bystander effect. So this is a smart way to deliver a very high cytotoxic payload to a cancer cell, and it offers precise targeting, cytotoxic effects, and minimizes off-target toxicity. The reason I'm talking about it is because there is a growing interest in looking at this class of drugs for patients who have non-small cell lung cancer, have an actionable alteration like we've talked about today, and unfortunately their disease is growing. And so look here, you can see some potential drugs that are already approved or already being studied in clinical trials. And there's different types of antibody drug conjugates. There's ones where we really need to know if the tumor is expressing a certain pathway or protein, so we can link onto it and cause that effect, that cytotoxic effect. And then there's some where we don't necessarily need to know how much of that protein is being expressed on the cancer cells, and we can still use it. And so these are potential off-the-shelf, ready-to-go drugs that we can reach for in the future or on clinical trial to help overcome that resistance that some patients may experience on targeted therapy. So stay tuned. We'll talk more about this, I'm sure, this session. So my summary and key takeaways: 1, resistance to targeted therapy is common and it's complex. 2, disease progression type and the extent of that disease progression really does matter. 3, and this is essential, please discuss it with your team. Post-progression testing is key. So that liquid biopsy, that tissue biopsy, advocate for yourself, talk about the pros and cons of that. 4th, clinical trials should absolutely be considered. If your center where you're being treated does not offer clinical trials, please ask your oncologist, hey, I'm interested in a clinical trial. Is there a center that's nearby that you might recommend? Sometimes they're the best source to offer that referral. And then finally, there is hope. There's hope for new therapies like ADCs, like new targeted therapies or TKIs that are emerging. Finally, I'll end with this. Patient support groups are so important. So groups like Cancer Grace are so meaningful. They offer an opportunity to partnership, to empower and to educate our patients. This is a list of all different biomarker specific groups as well that you can inform yourselves about, that you can get involved with and communicate and connect with patients who are in a similar situation as you. Thank you again for the opportunity, and I really appreciate it.
Thank you, Dr. Singhi. We'll leave the questions again to the end. And next, it is my pleasure to introduce Dr. Iyengar, who is a friend and director of the Metastatic Service and member of the Thoracic Services, the Department of Radiation Oncology at Memorial and Sloan Kettering Cancer Center. He's definitely a giant of a thoracic oncology. Many publications that he's going to cite today has his name on it, so that's kind of cool. And in addition to him being a clinician, he's also a basic scientist, and he has a lab which studies cancer cachexia. Go ahead, Puneeth.
Well, Ludmila, it's a wonderful opportunity to speak. Anytime I get a chance to speak for Grace or to patients and patient families through Grace is an honor. It's also an honor to follow Dr. Singhi, who has set the table wonderfully for this discussion. And so I'm going to talk about the role of radiation in patients with oncogenic drivers. As Dr. Basanova pointed out, I'm from Memorial Sloan Kettering Cancer Center. These are my disclosures. And so when we think about non-small cell lung cancer and outcomes for non-small cell lung cancer, I always, and have done so for about 15 years, started with biology. We all understand, whether we're patients, patient advocates, family members, scientists, clinicians, nutritionists, so forth, that biology really is critical for effectuating outcomes. You've heard it from a number of our colleagues today. And, this section is focused on targeted therapy, and certainly EGFR mutant positive disease, ALK mutation positive disease are critically important for driving outcomes. And what I would like to argue, and what Dr. Singhi was getting at, is that the role of the pattern of disease distribution, whether it's oligometastatic versus polymetastatic, the relevance of acquired mutations and resistance mechanisms across variant evolution is all going to play an immense and holistic role in how we treat all of you. And the reason that it's important is because systemic therapy is getting better. This is a chemo IO study, but it was actually predated by a number of years by very critical targeted therapy trials. But the important point to note is that we are doing better and better because of our medical oncology colleagues and our scientists, and of course our patients who took part in these trials, to improve systemic disease control with systemic therapy. But what I would like to always argue is that these treatments are not as durable as we would like. They don't have a depth of penetration as we would like. And so therefore, we want to do better. And so aside from improving systemic therapy, which I know is going to happen date by date, year by year, we want to see if other treatments can also play a role, local treatments. And when I talk about local treatments, it's both radiation and surgery. So why have we started talking about local therapies, radiation, surgery in the metastatic setting beyond palliation? Because for up till about a few years ago, we would only use these therapies for improving palliative symptoms, whether it's pain, bleeding, shortness of breath, etc. But as systemic therapies got better, we started thinking maybe there's a role for local therapy in improving survival outcomes. And then we still had a niche because these systemic therapies weren't perfect. They weren't curing or providing long-term durable benefits for all the patients. With imaging, we started finding metastatic disease earlier to be able to understand whether they could be benefited by targeting with local therapy. So we asked the question, can local therapy promote a situation where we turn disease that was once not salvageable into a chronic illness? And so we have a lot of evidence for that in other cancers beyond non-small cell historically. You take sarcoma patients that were metastatic to the lung or liver. You take colorectal cancer patients metastatic to the liver. These patients had these areas surgically resected. And so the surgeons were really at the forefront of showing that surgically removing some of these sites of disease was actually beneficial from a survival benefit. Then we started looking at our non-small cell lung cancer patients and those with isolated adrenal metastatic sites of disease that were resected, or brain mets that were resected or treated with SRS. Those patients started doing better and better. So really, we started thinking about when we would offer local therapy in terms of sequencing. Could we offer, and there's one other, and the first one I should mention here before these three is upfront. If you are diagnosed with metastatic disease, but you have only one side of disease in the brain or in the adrenal, and that's taken care of by surgery or SRS or radiation, why can't we be aggressive with local therapy for the remaining disease you have in your chest? So that's treatment up front at the time of diagnosis to go along with systemic therapy. There's not as much data about that, but I'll maybe provide some highlights later on in the question and answer part about how that can actually avoid some of the acquired resistance that we see in our patients. But the areas that I wanted to focus on are consolidation and oligoprogression that Dr. Singhi had pointed out before. Consolidation being patients receive X amount of systemic therapy. We want to make sure that the disease is still controlled, and then that's the time to offer local therapy. Versus oligoprogression, where patients are receiving systemic therapy for some definite period of time. They may develop acquired resistance, and then we treat whatever areas are progressing, allowing these patients to remain on the same systemic therapy in order to optimize their long-term outcomes. So unfortunately, or you could take it two ways. Most of the studies that we've done that have looked at use of local therapy in advanced non-small cell lung cancer patients have been done at consolidation and have been done in patients that do not have targetable mutations. So these are some of the major studies that were conducted over the last 10, 12 years that really started showing us that in a randomized small series of patients, those that received local therapy may actually have an improvement in progression-free survival and, in fact, overall survival. And the Gomez study, which is the topmost study that has a red arrow, did include a percentage of patients with EGFR mutated and ALK mutated tumors. And so we did have a signal that local therapy is good, it's safe, and it may improve PFS and overall survival. So all of those small studies culminated in a larger study. But this larger study, just as a reminder, called LU002, did not have patients with target mutations. We started off by focusing on patients that received immunotherapy-based systemic therapy. They got 4 cycles, as long as they had controlled disease and few sites of remaining disease. They were randomized to either radiation and surgery, followed by maintenance systemic therapy, or just maintenance systemic therapy. And this is the largest study ever in the oligometastatic non-small cell lung cancer space to ask the straightforward question: do patients with localized limited metastatic disease benefit from local therapy? And unfortunately, despite the increasing signals in the previous studies, there was no significant PFS benefit, no significant OS benefit, but there were lessons learned. And the lessons learned were as follows. It looked like the radiation and surgery did its job. There was fewer sites of disease that were treated with radiation or removed by surgery that failed locally. So that was really good. There was actually a trend towards a delay in the development of additional metastatic disease. So that was good as well. So if the local therapy was doing what it was meant to do, control disease where it was aimed, and delay the development of metastatic disease, why didn't that translate into a benefit in progression-free survival or overall survival? And what we've really come to learn, and this is going to be actually data that we're going to submit to a journal soon, was that toxicity played a role in this patient setting, in patients that were receiving immunotherapy-based systemic therapy followed by local therapy, that there was significant enough toxicity that it may have diluted the ability of us to translate a local control benefit and a delay in metastatic disease to a survival benefit. But look, we're learning our lessons, and subsequent iterations of these studies, we anticipate tightening up the treatment fields, reducing the toxicity, and potentially being able to show that local treatment in the form of radiation and or surgery is beneficial in a larger, type of study. But, so what were the limitations of these studies, a few of which that included targeted therapy? It's really that we're not doing enough to identify the patients through biomarker selection that are going to ultimately benefit from the treatment. And this schematic on the right is just an example. If you on the graph, if you start from the leftmost part of these curves, we treat patients with metastatic disease with local treatment in the hopes of improving their survival. But we, as radiation oncologists or even medical oncologists, have a difficult time predicting what direction your disease is going to go after we offer the local therapy. So at Memorial Sloan Kettering and other institutions, I think we're making a very strong effort to develop the biomarkers which are optimally going to select the patients that are going to truly benefit from local therapy. And the other thing I pointed out is we have to reduce toxicity. And those are really the key takeaways of that class of trials. But what about upfront therapy? What about targeted therapy? I think that the real benefits of local therapy will be in patients with targetable mutations. Why? Because their toxicity profiles are going to be significantly less, touch wood, and because there's a longer potential benefit with the use of targeted therapies in the setting of these patients. So this SINDOS study was a study that was conducted in Asia. And what they decided to do is to take metastatic patients that were starting on a TKI and then give radiation to all sites of disease and basically see whether it would improve survival. This is what we call the upfront treatment. And those patients that received both the radiation and the TKI had a significantly improved progression-free survival outcome. So basically, it gives us increasing signal that radiation is actually going to be actually beneficial. And MD Anderson has a number of these types of studies that are looking at the use of radiation or surgery and consolidation for these patients with target mutations, and we look forward to those findings as well. Hopefully Eric can give us some inside scoop on those things. But here, when I was at UT Southwestern before I moved to Memorial Sloan Kettering, we did a single arm phase 2 trial that's been submitted for publication to give you a sense of the types of patients that we included on the single arm phase 2 study. So it's patients with EGFR mutated tumors, and half of the patients had exon 19 mutations, half had exon 21. We had about 40 patients or so on the study. The average age was 65. Majority of the patients were female. But what was interesting is that we included patients up to even more than 5 metastatic sites of disease. So this included patients that had oligometastatic disease, that had what we call polymetastatic disease. And so we included all of these patients. And the idea was that these patients were going to start by receiving osimertinib. And then after about 2 to 3 months on osimertinib, we were going to treat whatever we could treat with radiation. And so these are some of the characteristics of whether radiation was offered or not, and why it was offered or not at the time of what we call consolidation. In the kind of fourth box from the top, or the second from the bottom, you see the types of sites that were radiated, including the primary lung, bone lesions, lymph nodes, brain, adrenal, liver, and so forth. And so this is just, by the way, a table of the number of studies that are a small proportion of the number of studies that are trying to look at patients with EGFR mutated tumors and to see if we can offer the local therapy in an optimized fashion to improve their survival. Again, part of the rationale is to identify patients that have metastatic disease that we offer the local therapy to before they develop this acquired resistance and may have disease that becomes more aggressive or progressive. And so these are the curves. It's a single arm study from these two study curves, but what you can see is that these patients probably did much better than the historical patients who were on TKIs alone. And I think this is a very illustrative study. What is the goal? What is additional to avoid resistance? The second goal is to allow these patients to remain on osimertinib, which is a wonderful drug, and is in many of these patients still controlling disease at a majority of sites. And so what you can still see is that a number of patients, even at the end of the study, remained on treatment on osimertinib, which is amazing. And we're talking about 4 years out. What we also found was that the arrows point to times where patients were given radiation. And you can see that a lot of the patients were able to be maintained on the drug for a significant amount of time after they were offered radiation, which is suggestive that the radiation and the local therapy did what it was supposed to do. So oligoprogression. Now, so we'll continue to talk about oligoprogression. And this is really why I think it's so critical for us to develop biomarkers. In the figure on the left, you see 5 patients that had the same EGFR mutation, yet they had widely disparate outcomes when it came to progression-free survival on osimertinib. One could say that there were many different reasons: the presence of different co-occurring mutations, the different host biology, meaning your immune system behaved differently, responded differently to the therapies. And so we are all, as a group, trying to tease out these issues. But it's also important for us to continue to add radiation in an oligoprogressive scenario where you've been on drug for a while and then you get local therapy. Remember, the previous patients all received radiation at consolidation, and a lot of them continued to receive radiation at progression as well. So it was almost like two situations. But ours was one of the first studies that was done in 2014 in collaboration with the University of Colorado. It was a single arm study that combined an EGFR inhibitor with radiation to show that it was safe and to show that it was potentially successful. And how do we say that it's potentially successful? These patients, back when we did the study, had very few subsequent line therapy options. They had failed multiple lines of therapy, but despite having failed multiple lines of therapy, their overall survival after our treatment was 20 months, and their median progression-free survival was 15 months, which was, at that time, we felt extraordinary. And it's, this is played out in randomized studies as well. And this is Dr. Tsai's study from Memorial Sloan Kettering, where they randomized patients who were oligoprogressing. Again, progressing in limited sites of disease. They randomized them to radiation versus continued systemic therapy, subsequent lines. And the patients that received the radiation had a better progression-free survival, had a better overall survival, and it was actually correlated with these line graphs. I mean, these graphs with these lines that you see in the middle, i.e. circulating tumor DNA. So the circulating tumor DNA was giving us a prognostic and potentially predictive model of whether these patients would benefit or did benefit from their local therapy. So it's still a work in progress. This study that's looking at radiation and oligoprogressive non-small cell lung cancer patients is being tested in a much larger patient population that includes EGFR and ALK mutated patients, and we're really excited to see what the findings are of that. So with the last 2 to 3 minutes, I have a number of things that I want to just throw out to the audience and the community. It's that overall, we need to do a better job, as I said, of developing biomarkers that predict which patients with metastatic non-small cell lung cancer patients with target mutations will benefit from local treatment. We don't want to overtreat the patients, unfortunately, that are doing very badly on systemic therapy, where they're progressing widely early. We don't want to treat the patients that are doing so well on systemic therapy, for instance, patients on lorlatinib who are progression-free for years and years. Why even offer them radiation when they're doing so well and avoiding any potential toxicities? We want to find that middle ground, those middle patients where the systemic therapy is doing just enough to keep disease under control, but where local therapy may be helpful. And so how do we go about doing this? Well, it takes, we think, a lot of data. We need to identify how patients do on standard of care TKIs, look at their patterns of failure, and try to predict and prognosticate what medical factors, clinical factors, pathologic factors, imaging factors can identify the patients that are going to met out widely versus develop metastatic disease in a limited fashion. Once we're able to do that, and we are using AI and other tools to understand those associations. Once we do that, we can actually start creating trials where we're enriching for those patients who we think will most benefit from a local therapy approach. And so, the real questions are, which patients will benefit, how will they benefit, and in what sequencing of treatment will they benefit? And these are the types of studies that a number of institutions and investigators are trying to understand. Again, in a schematic fashion, it's like the Goldilocks thing. We want it just right. We don't want to overtreat patients. We don't want to treat patients that don't need treatment. We don't want to treat patients that don't need treatment because it doesn't help. But we want to, those patients, we want to make their disease more controlled for a longer period of time with the offer of local therapy. And again, we're putting in a lot of different types of data to help us understand the trajectory of metastatic disease and the utilization of local therapy in terms of timing. And it's all, again, I think what Dr. Singhi said, it has to do with acquired resistance, bypass resistance, and just kind of the evolution of a patient's disease as a function of time and therapy. And so I'm just going to end with this slide, which again, we cannot underestimate the trajectory of a patient's disease, but we cannot underestimate quality of life and toxicity as well, okay? And so those are both of the factors that we need to be able to develop markers for prediction and prognostication in order to optimize our local therapy approaches with radiation. And I'm going to stop there and be happy to take questions. Thank you very much.
Perfect. Thank you, Dr. Iyengar. Can I have Dr. Singhi and Dr. Marmolillis show up? Hi, Eric. All right, so let's go ahead and start our panel discussion. So we have about 20 or so minutes to do that. Yes, about 20 minutes. So the first question that I have is, I'm going to start with Dr. Marmarella. So you talked about targeted therapy and you talked about toxicities of targeted therapies. Are there specific patient populations, such as elderly or comorbidities, where targeted therapies should be approached differently?
Yeah, it's a great question. I think the first thing that I'd like to address is how these drugs are developed. So when we develop a targeted therapy, one of the first things that is asked is what is the maximum tolerated dose? So at what point do we have too many side effects that that dose is too high? The question is not asked, what is the minimum effective dose? And so I think with a lot of these targeted therapies, the dose that is approved is likely higher than what we need. So I think the first answer to that question is that we can be pretty aggressive about dose reductions, or we can start actually a little bit lower and then start to make our way up in patients that we're worried about having additional toxicity. There are specific, patient populations that I think I worry about more. Certainly, the elderly, we would probably start at a lower dose if possible. But really, actually, I think these drugs are pretty well tolerated, and it's mostly dependent on which targeted therapy, whether I decide to start at a low dose and then move up.
Yeah. And the second question, I will leave it to pretty much anybody who wants to answer. Other instances where chemotherapy might still be preferable to targeted therapy, even if your patient has a biomarker positive disease?
Yeah, I can start answering that question. So there are some targeted therapies or some biomarkers where actually we do recommend first-line chemotherapy, not necessarily with immunotherapy, sometimes with. So it really depends on the biomarker. So for example, with our KRAS mutations, KRAS G12C, for example, currently our standard of care is to give these patients chemotherapy plus immunotherapy. We look at other biomarkers, for example. So I would say it really depends on the mutation. So we don't always want to rule out chemotherapy. Sometimes we have to rely on chemotherapy if a patient develops resistance on a targeted therapy. So sometimes chemotherapy can still be our friend. It can be considered, but have that discussion with your team. It really does depend on the biomarker.
Yeah, and then thank you. Question to Dr. Iyengar. The common things that we have discussions with our radiation oncologists, like, in the case of oligoprogression, you're considering radiation, the patient, let's say, continues on, osimertinib or lorlatinib. Do we have to stop the drug? Can we radiate while continue targeted therapy? What is there special radiation locations where you want to stop preferentially versus others where, like, I really don't care, you can continue? How do you approach that discussion?
Yeah, that's an amazing question. And despite having done this for so long, we still don't have ideal answers. I mean, so, it's a much more simple question when you're talking about chemotherapy, for instance, right? I mean, if we're treating the spine, if we're treating the brain, if we're treating any kind of viscera, we want to avoid doing it in combination. I don't have necessarily those concerns with TKIs, with osimertinib or lorlatinib. What I'm more worried about, the synergy of each of the treatments with respect to pulmonary toxicity is, I think, probably my bigger concern. And so I would argue that if we're treating large field in the lungs, whether it's primary with mediastinal adenopathy or so forth, then I may ask them to hold off the EGFR inhibitor for a little bit of time. If it's SBRT to a small lung lesion or some of the other things I mentioned, I'm not really as concerned about. But I don't hold it against people who want to stop the EGFR inhibitor 3 days before and 3 days after radiation for SRS or for SBRT, because we just have limited data. But at least from the trials I've been involved in where we did not hold it, there was not any increased toxicity signal.
Yeah. And I think the next question, I'll still have you comment and maybe have Dr. Singhi as well, Dr. Marmarels get their opinion. Brain mets, right? So we've talked about brain mets as one of the resistance, localized persistent progression. We didn't talk about it in Dr. Singhi in your talk because your talk was about resistance, but a very common situation where we have a patient with stage 4 diagnosed with brain metastasis. Do they all need radiation? When, how do we decide?
Yeah, you know, it's when I first became a radiation oncologist or a resident, the moment you had a brain met, we had to treat it. And unfortunately, back then we were treating with whole brain. What I learned over the years is absolutely we do not have to rush to treat brain mets if they're asymptomatic, small, lacking association with significant peripheral edema, and now even more so in the era of TKIs and immunotherapy that have penetrance. If their lesions are relatively small and patients are asymptomatic, I don't treat them. In fact, we actually have a trial at MSK, and there are probably multiple institutions that are having these trials where patients that have upfront CNS disease, we're randomizing them to upfront SRS versus delayed SRS at the time of either maximal response or progression, and the idea to see if it really matters. But I don't think it's necessary to rush into the treatment.
And Dr. Singhi, Dr. Marmarels, do you agree?
I agree. Yeah, I think it really depends on the biomarker that's identified. We do have a lot of CNS penetrant, highly CNS penetrant targeted therapies that are now available, like osimertinib, for example. So I agree. I really take it into the context of the patient. We talk about the pros and cons of pursuing a local therapy to the brain. So at our institution, we actually have a brain metastases clinic. And so I'll often refer my patients at the time of diagnosis if they have de novo at the time of diagnosis intracranial disease, they'll have that discussion because with every treatment modality does come a potential for adverse events, for side effects, right? And this is more of a journey that patients have with their cancer, not necessarily a sprint. And so we have to think very thoughtfully about how we treat our patients and sort of what they're going to develop from a toxicity standpoint.
Yeah, and the only thing I'll add is I think the key to this type of approach is short interval follow-up. So if you're going to forego radiation, which is really a pretty sure bet that you're able to treat the brain metastasis and wait for the systemic therapy to see if that's going to work, you need to make sure you're doing a brain MRI, in 4 to 8 weeks so that you're not missing disease that might be harming the patient.
Thank you. Question to you also. I don't know if you were listening to the previous presentation, but Dr. Yoon showed NCCN guidelines where we have a mutation and then the list of drugs. And for some mutation, the number of drugs that's listed there is like 6, 7, 9. How do you decide which of the targeted drugs to use in the situation where more than one has an FDA approval?
Yeah, I think you made the comment earlier that the NCCN guidelines are confusing, which in this case is definitely true. So I think there are a few answers to that. The first is that as you get familiarity with these drugs and these trials, you have your preferences about when you use certain ones. And I think that that's been formalized in some ways in pathways within institutions. So some institutions have very formal pathways about their preferences. Others are a little bit more, they're not as formal. It's more of a group consensus. We generally start with this in this situation or this one in this situation, and we kind of all agree. I do think and I think people are going to talk about it in some of the next sessions, it's actually getting more confusing, and there are probably more opinions about which drug to start with. But one of the ways that we have tried to formalize it is that when we have an alert system for when we detect something that's targetable, and we put down the pen preferred options. It's often not just one. It might be 1 or 2 or 3, but it kind of gives a consensus to the rest of our health system providers about kind of what the group thinks about what to do next.
So next, I'm going to open the Pandora box, and I'm going to give that Pandora box to Dr. Singhi. So disease monitoring with cell-free DNA in patients with, we are in targeted therapy sessions with oncogenic drivers. Do we do it? Do we not? What's the benefit? What's the cons versus pros?
Wow, yeah, that's definitely Pandora's box. So my recommendation at this time is on clinical trial. Yes, absolutely, I support it. The problem is, and I talked about that concept of minimal residual disease, right? That level of detection of disease that's just underneath the scans where we can't see it. Our scans are only as good as they are with level of resolution of detail, right? How do we capture, how do we detect that MRD, minimal residual disease? Some of these tests that we have right now for cell-free DNA, liquid biopsies, if you will, don't have that level of sensitivity. So sometimes they may seem reassuring with a negative result, and then you have this false sense of confidence, and unfortunately there is disease brewing, right? So you don't know. Or it's the opposite, where it detects it at such a low threshold, and then you have this positive result. You don't see anything on scans. Your patients are doing well. Their quality of life is preserved. And now what do? We're supposed to give them chemotherapy, try to sign them up for more toxic drugs, right? We don't know the benefit. So my answer is yes on clinical trial. 2, we're learning about what to do. And 3, have that discussion with your team and sort of what your quality of life is, what your treatment goals are, and open it for dialogue.
Yeah. A second question I have. Oh, go ahead.
Oh, I was going to say something interesting. And I hope I don't in any way, no way do I mean to be like create ambiguity, but I still think we're still learning what all of this means. And the simple question I ask is the DNA that we are seeing in the blood of patients DNA that is from the dominant clone in a tumor? Is it DNA from a clone that's so dominant that it's growing so fast, it's outstripping its blood supply, and then that's why it's leaking? Is it from the cells that are responding to therapy? Like I still don't even understand, what is being represented in those samples. And so I think, we're going to eventually, and we're going to do it fast, develop the associations between what we're seeing and outcomes. But I think we as a group continue to need to understand what is being represented in those specimens as well.
That's a wonderful comment, and actually a segue to my next question. Post-progression biopsy in our patients who develop resistance to targeted therapy. That was actually a question from the last session, but I saved it for here because I knew we're going to be talking about resistance. Do we biopsy the lesion that's growing? Do we biopsy any lesion? Do we even biopsy? How do you approach your post-progression biopsy decisions in a patient who developed resistance?
I can start with that one. So I think in an ideal world, you would get a post-progression biopsy of tissue, and it would be of an area that is growing. Because we do know that there are subclones, and areas that are not growing may not represent the overall changes that other areas might see. So if that is possible, I think that is what I would do. I think the second best would be an area that is lighting up on a PET scan. So even if it's not growing, it's still very metabolically active. It's still an area that is trying to grow. That would be my second choice.
Anybody else have?
Well, I was going to say totally agree about both of those approaches. The other thing I plead, and it's easy for me to say, is that if patients are offered the opportunity to get or to take part in a study where they're getting longitudinal sampling of disease upfront before therapy and at the time of progression, please sign up for it. Because I think, and it's the same issue that I deal with in my patients with cancer cachexia, where we are trying to get specimens of fat and muscle, and it's superficial specimens, but patients are going through so much that they just don't want to think about this extra stuff, and I totally get it. But I will tell you that the more sets we get in the same patient as a function of time, the before and the after, will give us much greater clarity about what is driving the progression, the increased FDG avidity, and it will really probably give us some insight into what to do next down the line. So again, easy for me to say to take part in these studies, but I think if you do and you have a chance to do so, it will provide some incredible insight, I think.
And since I have you on the podium right now, I'm actually going to pull one of the questions from the Q&A session because this is a question that comes up pretty commonly. So if oligoprogression is around 3 of your spots and those are treated with radiation, what if you get oligoprogression again, let's say a year later? Can you continue radiating those spots, or not? What's your limit? And that's a question to you. And the question to Dr. Marmorelos and Dr. Singh is, when do we switch systemic therapy, right? Oligoprogression, radiate, continue the same. Second oligoprogression, radiate, continue the same, or switch. How do those decisions are made?
So to answer your question, as radiation oncologists, we could keep radiating. And so it's technically feasible to continue to radiate. We don't have to use the same exact dose or total dose or dose per fraction, or number of fractions, but we can continue to administer meaningful tumoricidal doses or ablative doses of radiation. I think the bigger question is, should we do it? If I may just real quickly answer what I think about this, it's decision points. And so I've been thinking more about oligoprogression and progression beyond our treatment beyond progression a lot lately. And I've broken it down into decision points. And each imaging set that you get is a decision point for medical oncologists. Do we switch, not switch? So we looked back at our series of patients over 5-6 years at MSK, where each of them had either were treated with the ALK inhibitor, EGFR inhibitor, immunotherapy, and we looked at each decision point, and we tried to map out the burden of disease, the anatomic distribution of disease, and how or why medical oncologists decided to switch or not switch. But I will tell you that at least at this one academic institution, their instincts were spot on, because their decision points not to switch portended to better overall survival outcomes. And so it goes back to this idea that if you could keep patients on their first line of therapy longer, you usually are going to do better for those patients. And so, and by the way, number of METs is, I don't think any longer meaningful, because it's really about the velocity or trajectory of the disease. Because in the future, I have this prediction that a patient may come up to me with 10 METs, but if we can say that we can treat all 10 METs with local therapy, and then they'll have 2 years of a disease-free interval, we may go ahead and treat those 10 METs. And the opposite may be true, is that if we get a patient with 1 MET, and it should be a slam dunk to treat that 1 MET, but we know that on standard of care systemic therapy, they're going to have 15 more METs, then it's not relatively beneficial to treat that 1 MET. So I'm hoping that over time we go beyond the number of METs and just the trajectory as our means of decision analyses. And sorry for the long-winded answer, but I'll leave it to our medical oncology colleagues.
I like how you summarize that. I like how, one, you said these decision points for scans. That's a really big opportunity. We talk a lot about the scanxiety concept, right? Where patients are really nervous leading up to the scans, but so are medical oncologists. We're also worried too about, are we going to have that difficult conversation and lean in? Or are we going to be able to say, things look good, we can stay on the same therapy? So I like that idea of being able to continue on the same therapy as long as we can. That means we have more options to pull for the future, more options to sequence, and opportunities for clinical trials for patients. So that's a big priority of mine. What I highlight as well, too, is how quickly the time frequency between oligoprogression or development of new metastatic disease, how quickly is this happening and how often is it happening, right? So if it's happening quickly, if it's happening often, yes, we can keep referring back to our radiation oncologist, but if that frequency is quite high or the time interval is quite low, then we maybe should be talking about considering a change in systemic therapy. And then it also depends on where the progression is happening, right? If it's in a precarious area, an area of high concern, we need to think about, if it's time to change systemic therapy. So that's sort of my general thought process.
Yeah, I think the only thing I'll add is, certainly it's a marathon in terms of trying to string together different therapies. And so if at all possible, I don't like to make 2 changes at once. And so if we're going to do radiation, I try to keep patients on the same systemic therapy. If we're thinking about radiation, but we want to change the systemic therapy, I'll actually make that change first and then wait and see if we still need to do radiation. So if possible, I like to make 1 change at a time.
Mm-hmm. Question to Dr. Marmorelos. I'm pulling it from the Q&A session. How long does it take for the resistance to a targeted therapy to occur?
It's a great question. It's very patient specific. And we have some general medians from clinical trials about how long until a patient may have disease progression on certain targeted therapies. But that's just a median. 50% of people have it, earlier than that, and 50% have it later. So it can be quick. It could be a matter of months, or it could be years.
Another question that I'm going to pull, thank you. Another question I'm going to pull from the Q&A session, I'm going to address it to Dr. Singhi. When you were talking about resistance to targeted therapy, you mentioned the chemotherapy is one of the options. And the question from the audience we have, can chemo reset the cancer?
Yeah, that's a good question. I typically don't describe chemo as resetting the cancer. What I say is that it's helping to treat the cancer, right? And so I continue to go back to this. We're always trying to do better than chemotherapy. We're trying to be more precise, but sometimes we need chemotherapy to really get that response. It can work quickly, especially for patients that have a transformation to small cell lung cancer, and we know it's effective. So I don't typically say that it resets the cancer. What I say is that it can augment sort of treatment, augment symptom support for patients that are experiencing disease that's causing issues.
I think, by the way, I was going to say, Ludmila, I was going to say the patients or the audience was asking extremely phenomenal and insightful questions, because I've had this question for the longest time, is that if you take a patient who was doing well on a TKI for 5 months versus one who's doing good for 18 months, and then you offer both of those patients chemo after, will they get the same benefit from the chemo? And we looked at that data actually, and the answer is no. It's the patients, at least from our our data set, the patients that did better on the TKI ended up doing better on the chemo. And so who knows what that means, but I mean the questions are pretty amazing actually today, very insightful.
Yeah, and I think building in what you say, in your area of expertise, and I think those patients will also gonna do better with radiation, right? So if we take them 2 years to progress,
Right.
we know it's you were talking about the biology of the disease. We know that this is not the same biology as a patient who has, let's say, a primary progression
Yep.
on targeted therapy. So definitely plays into a role. Another question I'm going to also pull from the Q&A session, because I think the Q&A questions are excellent. So we've talked about, importance to get a post-progression biopsy, and sometimes it kind of just makes us adjust our treatment. What if your patient is progressing in the CNS only? You really cannot easily get the tissue unless you're resecting, and we can talk about, is there a situation where we go ahead and resect the brain met? How do you test for the new mutations in this situation?
I can try to answer that. I'm typically not testing. I think that's a really difficult area to biopsy, to be honest with you, because every decision point, like Dr. Iyengar said, is critical. And so we need to think about what's the pros and cons of going after an area where it's really tough to biopsy. That being said, I do think it's really important to get another liquid biopsy, right? If there's not an opportunity to have a feasible tissue biopsy. Tissue biopsies are gold standard because we can not only see and appreciate some of the acquired mechanisms of resistance that I talked about on target, off target, but that's the only way, right? If we get a tissue biopsy to see if the cancer cells themselves have changed to something like small cell lung cancer. I can't even tell you the number of cases I've gotten from different hospitals where the patient at time of progression, being on a targeted therapy, went and got a liquid biopsy. Liquid biopsy didn't show anything meaningful, but then we went and got a tissue biopsy and we learned, hey, this actually changed from non-small cell lung cancer to small cell lung cancer. That's a huge change to appreciate. So my answer in a long about fashion is to say it's not always feasible to get a tissue biopsy. Advocate for a liquid biopsy. And if you get a liquid biopsy only, sometimes that doesn't solve it. So a lot of nuances to appreciate.
And we've talked about brain CNS, and there's certain things we do for that CNS progression. When you look on ex-CNS progression, do you look at them differently if it's like liver, adrenal, bone, or it's not the site that makes you do different decisions, but rather the number and the number of the progressing sites?
I think, I mean, just the secondary analysis we did on that LU002 trial where we consolidated, the patients that had liver mets did worse. And that's not surprising. I think people have thought about that once disease gets to the liver, it's going to be a little bit difficult or more challenging. But it doesn't mean that it's the end of the road. I mean, I think that we just have to come up with, other options. And some people, there's a trial here that we're doing where, we're aggressively radiating the first signs of any liver disease from metastatic non-small cell as a means of trying to change the trajectory of the disease makeup. And so, yeah, I mean, I think personally, the presence of disease in the organ is probably more critical than how much disease is in the organ. But I mean, these are all things that we could figure out with the data sets that we're looking at, but I still don't have a lot of insight into that. What about you guys?
Well, I think if a patient's on immunotherapy and they develop a disease in the liver, I am more concerned about the effectiveness of immunotherapy and would certainly advocate for that patient getting chemotherapy or changing the chemotherapy. For patients on targeted therapies, if we're thinking about additional targets, I think targeted therapy is actually pretty effective in the liver. So it's different for each patient scenario, but I agree that the site is not as important in terms of choosing the next therapy.
Thank you. I'm going to pull the next question also from the Q&A session, and it's really nothing to do with targeted therapy, but I think it's such an important question that we need to address it. And I'll address it to Dr. Sankey because of your work with patient education on clinical trials. So who pays for participation in a clinical trial? Is it the patient insurance, or does the clinical trial pay for the drugs, scans, administration? Because I do hear concerns that maybe I cannot afford to be in a clinical trial. If you can address that.
Yeah, that's a great question. And unfortunately, it's a barrier that we need to continue to overcome. We really need to advocate as providers and as patients, as patient advocates, that access matters in different ways. So not only access to the drug, but also just physical barriers, right? So logistics to transport and travel yourselves, to stay at a hotel, to get to the center in the mornings, to leave after a long day, and to have that caregiver also be supported. So typically, the first question that I would ask is my insurance in network, right? To be able to obtain the treatment on the trial. And oftentimes financial clearance and the department at that institution will run that immediately. So you can find out if you're in network to receive the drugs. But the second thing I would ask the clinical trial coordinator or the nurses to say, does the sponsor of the trial offer just additional funding to allow for myself, for my caregiver to transport, to have lodging, things like that. There are some trials actually, and a lot of pharmaceutical companies now are starting to step up because of patient advocacy groups like Cancer Grace and other biomarker groups, and they are offering funds. So ask those two questions, right? Is my insurance in network? And then are there additional funding mechanisms to support travel and lodging if you have to get to a facility that's not in your hometown?
Thank you. The second question, I think we've talked about it a little bit, and that's from the Q&A session as well. But I think we need to address it a little bit more. So, Dr. Inkari, when you talked about radiation, you mainly talked about oligoprogression, and you did mention the CINDUS trial. And I kind of want to pull everybody's what's the standard of care in your practice for not oligoprogression, for oligoconsolidation. So for our patients in the room, so those are the two terms we use. So oligoprogression, we're waiting for the cancer to grow and then we radiate the growing site. Oligoconsolidation is where we give therapy, we wait for everything to shrink, and we radiate things that we still see. And what's your practice for oligoconsolidation therapy in targeted oncogenic mutant patients?
Yeah, well, I would be hypocritical if I said I'm continuing to do this based on the results of my own trial for patients who are receiving immunotherapy. What I will say is I still think it's a legitimate option for patients on TKIs to consolidate. I think the data from MD Anderson, the data from UT Southwestern and City of Hope is just going to make things stronger and stronger. And Dr. Haymak, who's Dr. Singh's boss, has been thinking about this for decades, and really gets the idea that you want to hit these lesions before they reach the point of resistance translating into progression. And so I think that I still think that's the optimal time for patients who are on TKIs. And just a little bit broader, the patients on immunotherapy, I don't think it closes the door, but it prolongs it. So what I mean is that I would like to see the patients on more than just 4 courses of immunotherapy, maybe 7, 8, 9, to show that they don't have the proclivity to met out. And then, and if they're doing well and, they have some smoldering residual disease, I'm happy to consolidate.
Eric, Amelina, what do you do in your practice?
Yeah, so this is a question I'm starting to get a lot. So the majority of my practice has now become patients 50 and younger. So I'm seeing a lot of young onset lung cancer. Many of these patients have oncogenic driver alterations like EGFR, ALK. They're young, they're fit, they're highly motivated to figure out what's the most aggressive potential option for them, especially if they're having a good response to their systemic therapy. As our systemic therapies get better, as we're seeing younger patients who are more fit, have less comorbidities, this is a question that's critical. And I think that's a patient group where it's very valid to go and talk to your team, to seek out a second opinion and ask, can I receive local consolidative therapy? And so myself, Dr. Mara Antonov, who's a thoracic surgeon at my institution, are seeing many patients who are asking, is this an opportunity? And I think it's a fair question. Puneeth, you very nicely highlighted some of the data that we have, phase 2 studies, some phase 3 studies. And I really think if you are on a targeted therapy, if you've achieved a good systemic control, asking the question is a very valid one. And I think it's a very case by case individualized discussion.
Yeah, I think I'll just echo the patient selection aspect of this. I think it's both time on therapy, what that therapy has done for the patient, and then also at the time that you're considering local therapy, making sure you're doing a full evaluation, a PET scan, an MRI, so that you're not missing any sort of disease that might be growing. And you're really sure that this is the only thing that's left. Maybe it's even not very avid. That's the type of patient I think that potentially could do really well with this.
And when you make a selection of your type of local consolidative therapy, surgery versus radiation, what do you usually go with?
I think the vast majority of the time we do radiation. It's non-invasive. There's less downtime if you're trying to keep somebody on a systemic therapy in terms of healing. But we will consider surgery for sure in certain situations, especially in the lung. I think that's one area that we would definitely consider surgery.
Yeah, I mean, and Dr. Singhi can, he has a great team with Dr. Antonov and company. But, I think our surgeons will, if it's early in the disease spectrum, like because remember, these are marathons, as we've all talked about. Like, I think there's much greater appetite for doing surgery early. And then, if they develop metachronous metastatic disease or metastatic disease sequentially later on, then maybe more amenable to radiation as a local therapy. But I think, isolated lesions to the adrenal can be resected. I mean, to speak on behalf of the surgeons, of course, isolated CNS lesions could be resected, lung lesions. Yeah, so I think there's ample opportunity for both. But it needs to be really integrated into a multidisciplinary discussion. And that's one, we have guidelines for this, as does Dr. Antonov now from a surgical perspective. But the number one thing I think is common in both guidelines is that the patient's decision-making and determinations have to be first up front and center to the discussion. All too often, historically, we've made decisions and then come to the patient and said, this is what we think. But the patient's thoughts have to be really upfront in all of this, as Dr. Singhi pointed out.
Yeah, I'll echo everyone's comments. These are probably my longest discussions in clinic now that I'm having, and it's a good conversation to have, right? Patients with stage 4 disease are having an excellent response to systemic therapy, and they're asking, what else can I do to be able to stay on the same drug and have a meaningful response in time with my loved ones? And so we ask that question all the time. They'll come and they'll meet me, they'll meet the radiation oncologist, and they'll meet Dr. Antonov typically, and they'll have long discussions. Puneeth, I'll throw a question back at you because I get this question very often. I'm curious what your perspective is. Some patients will ask, if I go for radiation now, like to the lung, if there's some disease I want to consolidate, but then later, I'm thinking about surgery. Have I burned my bridge if I get radiation first? Does it make the surgery hard? Does it matter with sequencing?
Yeah, I mean, I think it does make the surgery harder. There's no two things of saying it. Radiation does cause inflammation that leads to fibrosis. Back in the olden days when we did radiation and then they did surgery for the neck, they used to say, oh, if we're cutting through concrete. Things are much more focused and specific now with radiation, so you don't cause that. But there will be times where it does make the surgery more difficult. But what I've been told is, the previous situation where we're doing a lot of radiation before surgery was superior sulcus primary lung cancers, and we were doing chemoradiation followed by surgery, and those patients got through it. So again, it's a good discussion to have between the radiation oncologist and the surgeon, is that if for some reason they want to do radiation first, make sure that maybe a dose and a fractionation that may not and a distribution of disease that may not portend to a more difficult surgery down the line.
Question to Dr. Singh. So when you were talking about resistance to targeted therapy, and one of the resistance mechanisms you highlighted in EGFR patients is MET amplification, or any things with MET. Why can't we just give everybody MET in the beginning together with osimertinib, since we know this is the most common resistant mechanism? Why is this not currently considered a standard of care?
So actually, it is kind of an option, right? If we think about it. So now we are talking about, patients should know, I just did a video on this recently, that there are now 3 FDA-approved options for patients with EGFR positive stage 4 disease. And I mean classical EGFR mutations, exon 19 deletion and L858R. Patients should know that we have the tried and trusted single agent osimertinib, which many of us have leaned on since the approval in 2018 in that setting. But now we also have what we call combination strategies, where we're trying to do better, right? Trying to prevent some of these mechanisms of resistance that develop. And so the combination that I'm referring to, where we've actually seen that if patients start off on this combination strategy, they're less likely to develop MET amplifications, is with the strategy of a drug called amivantamab, which is a bispecific antibody that targets EGFR, but also that MET pathway. And then we're adding in a 3rd generation EGFR targeted therapy called lazertinib, which is somewhat similar in mechanism of action to osimertinib. So yes, it's a very valid question. We're trying to learn and understand if we can use combination strategies to prevent those mechanisms of resistance from developing that might be common.
Good. Question I want to ask Dr. Marmarell is, in lung cancer, we sometimes, let's see, 2 different tumors, one in the left lung, one in the right lung, and nothing in the middle, and you do the molecular testing on one tumor. This is, let's say, EGFR mutation. You do molecular testing on another tumor, and it doesn't have an EGFR mutation. What does that mean for you, and how do you approach that this patient?
Yeah, so I think molecular testing has brought this to our attention probably more than we would have seen otherwise. And this happens. And it is possible to have 2 early stage lung cancers that are detected at the same time. So in the situation you're describing, where one has an EGFR mutation and the other doesn't, I would treat each of those as though they are their own primary lung cancer. So depends on the size and the location and all those things, what you would end up doing either for local therapy or systemic therapy. But it is possible to consider those as individual early stage cancers.
Thank you. A question I have to Dr. Iyengar. Can you talk to us about protons? It's a common question that our patients are asking, like, when do we should do protons for the lung radiation? When should we do the traditional electron? Can you just describe your thought when you go through that question?
Yeah, sure. So so protons and the traditional radiation we use are 2 different forms of radiation. So the traditional radiation we use are basically very powerful X-rays. They're literally X-rays that you would have for a chest X-ray, or if you have a fracture, but they're much higher energy. They have a very great depth of penetration, and we focus those X-rays conformably to where you have disease. The issue technically with those X-rays is in and out, meaning a lot of it gets deposited where the tumor is, but there's still some exit dose beyond. And when we were treating stage 3 lung cancer patients a number of years ago, we had to deposit the radiation somewhere. We had to plan to deposit those X-rays somewhere, and that somewhere would often be the heart. And it was the heart because unfortunately back then those stage 3 patients weren't living long enough for us to see heart findings. Now they are. And so now we've had to readjust, where that radiation gets deposited and directed. Protons, another form of radiation we call more particle-based energy. And these are like little individual particles that have a direct effect on the DNA damage, the DNA by directly damaging the DNA. Whereas X-rays knock off electrons from tissue, and those electron changes in densities affect downstream DNA. So the advantage of protons theoretically is that when we deliver protons, the radiation stops, meaning it stops where the disease is. There's no very minimal exit dose. So theoretically, there should be a lot of normal tissue protection. Where do I think protons are amazingly important for? Pediatric patients who are going to live a very long time, increased likelihood of secondary malignancies 30 or 40 years down the line. Patients with CNS disease, patients with skull-based disease, patients with spine disease, areas that are very sensitive to radiation and have, limited options about where radiation can be deposited. With respect to lung cancer now, let's go to lung cancer. There's not been a whole lot of data to suggest that protons are far better than X-rays, especially with our most modern techniques that we use for avoiding normal tissues. Where it could be important, again, is disease that's right next to the heart, where it could be is disease that's very close to the esophagus that the protons can benefit. We're starting to think that maybe large swaths of blood in vasculature or bone limit patients receiving subsequent systemic therapy because of toxicities down the line, because we've affected blood cell development. So there's actually a randomized study that was just completed in NRG comparing patients receiving X-rays, like high-level competency X-rays with protons, and for locally advanced lung cancer. So we'll see if it benefits the lung cancer population. I think more than likely there's not going to be a whole lot of difference. So long answer is that for lung cancer, protons don't seem to me to be the kind of the most obvious approach in most settings.
Thank you. Another question that I want to address, it was actually asked in the first session, but we didn't have time to go through the session. So our discussions about targeted therapy and resistance is a moot point if we do not perform the molecular testing. And the question that I have is, what, in your opinion, are the biggest barriers to the widespread adoption of comprehensive molecular testing in lung cancer? And taking into account that we are talking to patients, what can patients do to make sure that molecular testing is performed on their tissue? Anybody?
I can start on this one. So there are a number of barriers. The first is that the tissue that we get from a biopsy may not be enough for molecular testing. And some places are not sending both a liquid biopsy and tissue molecular testing because they're worried about insurance not covering both tests. So while that's ideal to be able to send both of those at the same time so that you can try to get an answer in a timely fashion, that's not always possible in all scenarios. The next barrier is that there actually are a number of options for where to send the molecular test and how to send it. And for people that treat a variety of cancers other than lung cancer, it can be kind of confusing about which is actually a comprehensive test. So we have, there are too many options, and the education around which options are adequate is still lacking. The next barrier I would say is at the beginning of diagnosis, there's a lot going on, right? So patients are having symptoms, you're getting scans, and the provider has to think to send the molecular test. They actually have to physically send the test, sometimes fill out the form, and there can be a delay because of that amount of time that needs to happen. So I think what patients can do is they can ask about their molecular testing. So they can ask to see the report. They can say what kind of molecular testing was done. Was it done on my tissue and the blood? Could we do both if something wasn't found on the one that was sent? It's a tricky situation. Thankfully, it's getting better. It is getting quite a bit better, but I think patients can ask that question of their care team.
Any other recommendations for our patients?
Yeah, I think you did an excellent job summarizing it. The International Association for Study of Lung Cancer, ISLAC, actually just put out a biomarker survey trying to understand why isn't every patient with non-small cell lung cancer getting molecular profiling. That is a huge call to action. It needs to happen. If it can't happen from the provider level, patients need to ask. Their loved ones need to ask. If you have non-small cell lung cancer, did I get molecular testing? It's also called biomarker testing. There's a lot of different names for it, but ask. And the other thing I'll just add on to what Melina said very nicely is it doesn't even matter what type of non-small cell lung cancer you have. I know in NCCN guidelines, they say non-squamous, it's recommended and you need to do it. In squamous, it says consider. No, I tell every fellow that comes into my clinic, even if you have squamous non-small cell lung cancer, you need to ask for molecular testing because I can't tell you the number of times I found an EGFR mutation. What if it's a combined histology, not just squamous, but adeno, and the piece of the biopsy just captured the squamous and you missed the adenocarcinoma? So get that testing.
And then the last thing I'll add to what Eric said is that if everybody views imaging as critically important to decision making, you have to view molecular testing at the same level as imaging, and understand the inconvenience, but it's equally necessary in terms of the right treatment decision making.
Thank you. And I think we have 2 minutes left, and I'm going to do one brief question to Dr. Singhi. Can you go over the definition of minimal residual disease again? There was a question from the patient, not clear on what that is.
Yeah, I mean, it's a good question. It's one that we're still learning a lot about, right? How to detect it. So really, the only way we can tell if a treatment's working or if we're running into trouble is two things. One, I mentioned symptoms, right? If a patient's not feeling well, that's a really bad sign. But the second thing we really rely on right now as standard of care, the way we all practice at an academic center, at a community center, is our scans. We use CT scans, we use PET scans. It can vary case by case and with discussion. But our scans are only as good as the level of detail of resolution. There can be some small amounts of disease, which we call minimal residual disease. If you have metastatic disease, stage 4 disease, sometimes you can have what we call micrometastatic disease, very small amounts of your cancer still present in the blood. And so what we're trying to do is to increase our capacity to detect this small amount of disease that may remain, that may be lurking, that we can't see on a scan. And we call that minimal residual disease, MRD. And so we're trying to advance our sciences with liquid biopsies, with circulating tumor DNA or cell-free DNA to say, can we capture the disease we may not see in a scan early? And can we do it in a different non-invasive mechanism like a blood draw? So stay tuned. I know the technology continues to get better. Penn is leading a lot of amazing things in this space. So are other colleagues internationally. But my hope is that we'll continue to get better and we'll be able to detect that MRD.
Thank you. All right. So I think with that, we have a minute left. I would like to thank our panelists for dedicating part of their Saturday to our patients. I think also I'd like to thank the patients because the questions for this sessions were really astute. And I think it made the session run a little bit faster, and we actually managed to answer pretty much all of them. So now, again, thank you, Dr. Marmarellis. Thank you, Dr. Einhard. Thank you, Dr. Singhi. We are now moving into a third part of our morning or afternoon, depends on where you are, where we're going to be talking specifically and covering each mutation that we briefly discussed above. And to start us with, it's going to be Dr. Christine Bestuina, who is a thoracic oncologist at the University of Chicago Medicine. She is interested, her research interest is synergy between immunotherapy and radiation, and she is very active in Alliance, which is one of our largest cooperative group, where she does clinical investigations to improve care of our patients with lung cancer. Go ahead, Dr. Bestuina.
Thank you so much. And thank you everybody for spending your Saturday here with us. Today, it is my job to talk about 3 specific mutations, so ALK, ROS1, and RET. And we're going to talk a little bit about pushing the boundaries of targeted therapies. Here are my disclosures. And so today I'm really going to try to focus on what I consider to be today's best targeted therapy available for each of these mutations. You've heard from several of my amazing colleagues earlier this morning about some of these mutations. They've been mentioned by name. Some of these drugs have been mentioned by name. It can be really easy in this space to get lost in 10 different clinical trials and options. So I've chosen to really try to focus on what I consider the standard of care based off of what data we have available. So we're going to start first with ALK. And so ALK inhibitors have really been available to patients outside of clinical trials for about 15 years now, since the first approval of crizotinib. Since that time, we've also seen approvals for ceritinib, alectinib, and brigatinib, and then most recently, lorlatinib. And we've had some time for some of the lorlatinib data to mature, and so we're going to take a look at some of that updated data of lorlatinib. This was presented by Dr. Jessica Lin at last year's ASCO. So one of the things that we talk about quite a bit for different TKIs or tyrosine kinase inhibitors is what generation are they? And when we use those terms, really what we're referring to is what's captured down on the bottom in this triangle. So with each new generation of tyrosine kinase inhibitors, we expect to see increased potency, meaning increased activity or getting in and blocking whatever that mutation is at a better rate. We expect increased selectivity. And what we mean is that it's avoiding some of the other normal tissue and hopefully decreasing toxicity that we can see with each generation. These TKIs are designed intentionally to get into the brain better. You heard a lot about that in the last session about the importance of coverage of the brain space, of treating brain metastasis, and the ability to delay radiation to many of these spots of cancer in the brain. And then additionally covering some of the resistance mutations that can occur. And so we know that when we use TKIs, new on-target resistance mutations can develop. And what that means is if you are on osimertinib, which is an EGFR TKI, you can get new mutations on EGFR that can outsmart that osimertinib. By covering a larger number of those on-target resistance mutations, typically we can either treat them if they've already developed or prevent them from developing in the first place, which I think is really the ultimate goal, to get the best drug to the most patients earlier. And so this is the CROWN trial. This looked at patients who have ALK-positive non-small cell lung cancer who had not received prior treatment for metastatic disease. Patients were randomized in a 1-to-1 fashion to lorlatinib versus crizotinib. Of note, we would not really consider crizotinib an acceptable control if it were to be designed today because so many drugs, particularly alectinib, ceritinib, and brigatinib, were known to have better efficacy and better CNS penetration, but this is the trial as it was designed at the time that it was designed. Importantly, this trial did one great thing. Well, it did many great things, but it allowed for patients who had asymptomatic treated or untreated brain metastasis to be enrolled. And this is something that myself and many of my other colleagues who are talking today have really been championing at many clinical trial design meetings, which is if we think drugs are going to have CNS efficacy, or if we know from prior experience that they do have CNS efficacy, allowing patients to go on without prior radiation is going to give us a better real world population and hopefully allow many patients to avoid radiation in the future. And so you've seen a few of these different plots already. Essentially, this just shows the percent of patients who are progression-free survival at a specific time point, meaning that their cancer has not grown, it has not gone new places in the time that they've been on the drug. And so at 5 years, 60% of patients had no evidence of progression of their cancer. And this is quite remarkable. I think I was fortunate enough to be in the room at the time that this data was presented. And this is really quite a significant advance over what we have previously seen for patients who have ALK. This is time to intracranial progression. So again, this is for patients who did receive serial MRIs of the brain, looking at if they already had brain mets or even if they didn't. How long did it take before any cancer grew or developed in the brain? And importantly, at 5 years, 92% of patients had no evidence of cancer progression in the brain. And for my patients, I know this is incredibly important. Your brain is your thought process, it's your personality. It's the most important real estate in your body. And so being able to protect that brain space and to do it for such a prolonged period of time is a really important movement in this field. Now, what about toxicity? And so I will say that there is more toxicity with lorlatinib than with alectinib, which previously had been my drug of choice. But as we use lorlatinib more and more in the front line, I think we just need to get better about managing these toxicities. So hypercholesterolemia and hypertriglyceridemia are the most common. I coach my patients to expect that they're going to be on some sort of statin medication. About 80% of patients end up on a statin when they use lorlatinib. And then weight increase is one of the big side effects of this drug that's been newer for us in learning how to manage. And so grade 3 weight gain is quite a significant amount. It's over 20 pounds of weight gain, which all of us know really can affect your self-image. It can affect your ability to do physical activity. For many of these patients, as Dr. Singhi has been referencing, they're young, they have families, they want to be active, they want to be able to exercise. And so this weight gain can be quite significant. And there's a lot of questions about, can we use GLP-1 agonists to help moderate some of this weight gain and to prevent patients from experiencing such high-grade toxicity. One thing that was not included in this presentation was the neurologic toxicity that can occur with lorlatinib, which can be significant. And it can be anything from just, I don't think quite as clearly as I normally did, to patients who have hallucinations. And so that's where getting a history from a beloved family member or care partner can be incredibly important. Importantly, dose reduction did not impact the efficacy. And so this is, I think, one of the most important things that came out of that presentation was if patients are not able to tolerate 100 milligrams, we can go down to 75 milligrams, or even in some cases 50 milligrams, and the drug still retains excellent efficacy. And so I know for my patients, they're very, very hesitant to go down on the dose, even if they're having toxicity when the drug is working. But I think with this data, we can do that in a reassuring fashion and in a thoughtful fashion. So what's coming if we are using lorlatinib as frontline? We have some new exciting drugs coming, and I'm going to focus on NVL-655. This is an ALK TRK sparing tyrosine kinase inhibitor. Excuse me. with this drug, we've been able to decrease some of the toxicity that we see with the TRK side effects of lorlatinib and some of the other drugs, while still maintaining excellent and even improved penetration of ALK penetration. Here I'm going to show you some of the lung cancer data that's been presented. And so even patients who had received prior lorlatinib still had significant reduction in the size of their tumor. So 35% of patients met what we qualify as a response, even though the majority of patients, which are those who are on the bottom half of this curve, had some amount of shrinkage of their cancer. These numbers also looked quite good for patients who had not previously been exposed to lorlatinib. This drug looks quite tolerable in the early phase 1 trial. The majority of toxicity is liver test increases, constipation, a change in taste, and some nausea. We've been fortunate enough to have this clinical trial at University of Chicago, and I have to say I've been really impressed with both the efficacy and the tolerability of this drug. With that, I'll move to ROS1. So similar to the lorlatinib data that we just looked at, with some of the data that we've seen for repotrectinib, which is a next generation ROS1 TKI, to me, this would now be the standard for the large majority of patients. Here we do see some activity against TRK as well. And so this drug is available both for NTRK and ROS1. It was designed to decrease the potential of developing ROS1 resistance. Here we're going to look at 2 cohorts of lung cancer patients who are TKI naive. These patients could have had prior chemotherapy, so they didn't have to necessarily be truly treatment naive. But you'll see that the response rate here was 79%, which is very impressive. For patients who had had a prior ROS1 TKI, and here that included crizotinib, entrectinib, and ceritinib, the response rate was 38%, with the majority of patients having some shrinkage of their tumor, even if it didn't meet our criteria of 30% shrinkage. For patients who were TKI naive, there was a median progression-free survival of 36 months, or essentially 3 years, without any evidence of progression of the cancer, new spots, or growth of what was there. For patients who had previously been on a ROS1 TKI, that time was 9 months. And so one of the big lessons here, and that I think we've heard throughout the day, is we do want to use our best drug first, because these numbers, which translate into very significant life responses to these medicines, are the highest when the best drug is given first. This drug works really well in the brain. So patients who had brain metastasis had even though these numbers are small, these are only 8 patients who had brain metastasis. The large majority of them did see a shrinkage in those brain metastasis on this drug. And that was true even if they'd had a prior ROS1 TKI. The toxicity of this drug is quite real. And so about half of patients did require a drug interruption, meaning a brief hold of the drug due to some amount of toxicity. And about a third of patients needed a dose reduction of the drug because of toxicity. We're still waiting to see what is the efficacy of this drug if a patient does require a dose reduction. Dizziness is one of the more unique toxicities that we see with this drug. 62% of patients reported some dizziness. 13% required a pharmacologic intervention, such as meclizine, and 3% had grade 3 dizziness. And just as a reminder, grade 2 dizziness can really significantly affect patients' lives. Grade 2 dizziness can give moderate unsteadiness or a sensation of movement, and can even limit instrumental ADLs, or activities of daily living. for me, what that really translates to is independence. And that's something that consistently is expressed as one of the most important things to patients, is the ability to remain independent. And so with sequencing of these TKIs, this is where, again, we can visually see the argument that starting with an earlier TKI, then using repotrectinib, the total duration on treatment is still going to be less than what we see with repotrectinib. Taletrectinib is an additional TKI that's coming, that's quite promising. Response rate of almost 90% for patients who hadn't been on a prior TKI. Progression-free survival or time on drug, almost 4 years here. And importantly, the tolerability here looks quite good. So patients can experience elevated LFTs, diarrhea, nausea, vomiting. But in general, this drug appears to be quite well tolerated by patients. So I think where taletrectinib will play in the ROS1 field, I look forward to seeing its use. Similar to the other nuvalent drug that we saw, NVL-520 is showing very good responses even for patients who have had prior repotrectinib, even if they've had multiple prior TKIs. An excellent toxicity profile from what we see in the early clinical trials, some LFT elevation, some weight increase. And then lastly, we'll just talk about RET. These fusions occur in 1 to 2% of lung cancer patients. The mutations can occur in patients with thyroid cancer, colorectal, and CMML. Selpercatinib is an excellent tyrosine kinase inhibitor with RET activity. In patients who previously had gotten chemotherapy, we saw a shrinkage response rate of 64%. That goes up to 85% in patients who were completely treatment naive. And we also see good responses in the brain with selpercatinib. One of the big toxicities to know about is high blood pressure. Patients do need their blood pressure to be well controlled at the time of starting the drug. And then recently we saw results published from Libretto 431, which is a randomized trial of chemotherapy with or without immunotherapy versus selpercatinib. Selpercatinib significantly overperformed, and I know there was a question earlier about chemo first for all TKI, about if there's any mutations for which chemo should be given first. This is not one of those drugs, but I think it did raise a lot of ethical questions about, should this trial have even been performed in the first place, or based off of the phase 1 data, did we feel confident enough that selpercatinib was the right treatment of choice for patients? Pralsetinib is a similarly active RET TKI with just a different toxicity profile. Here we see more cytopenias. And so in conclusion, we've had some new and exciting TKIs that have been approved for all 3 mutations in the last few years. And we've got exciting new generation TKIs in development that will improve efficacy, limit toxicity, and also continue to improve CNS outcomes for patients who have spread of cancer to the brain. Thank you.
Thank you, Dr. Bestuina. So we have very small amount of time for questions, but there's one question that came in earlier in the first session, but I left it for you. If you have a RET fusion and you are living in the area where you do not have an access to selpercatinib or pralsetinib, would you try, the dirty or the multi-kinase inhibitors such as cabozantinib? Would you go to alectinib, which also have a RET mutation, or would you just stick with chemo?
For that case, I would stick with chemo. So we saw in the control arm 431 that patients had a progression-free survival of about a year with regular chemotherapy. I probably would not add immunotherapy. We know that it has limited efficacy for these patients and can increase toxicity if the RET TKI is given later. But I actually find carboplatin pemetrexed to be better tolerated than some of the earlier generation dirty TKIs.
All right, perfect. Thank you very much. Thank you for you spending our Saturday with us. And we'll move to our next presenter. Thank you, Dr. Bestvina. All right, so our next talk, let me get to my, next is Dr. Akash Desai, who is an assistant professor of medicine at the O'Neill Cancer Center, University of Alabama in Birmingham. He is, his area of expertise is thoracic oncology and experimental therapeutics, and he will be discussing with us the MET, NTRK, as well as NRG. Go ahead, Dr. Desai.
Thank you, Dr. Bazanova. Can you just make sure you're hearing me clear?
Yes, we're hearing you very well.
Perfect. Thank you so much. And really excited to be here with everyone. And thank you, Cancer Grace and Dr. Bazanova and the team for inviting me to talk about this. So as was mentioned, I'm going to be talking about MET, NTRK, and NRG1 driven non-small cell lung cancer. And I'll take some of the parallels from Dr. Bestvina's talk, as well as some of the discussion that has been ongoing this morning in terms of NGS testing and such. So we'll move on with the first topic, which is the MET. And I want to just introduce a little bit about what MET is. And I think, often in clinics, and I'm guilty of this, we kind of start rambling all the genetic oncogene-driven non-small cell lung cancer mutation names, like everyone understands it. And so I'm hoping that, throughout this talk, I can try to break it down and kind of make it easier to understand. So we give all these fancy names. So MET stands for, has a scientific name for, MET. This is, essentially a gene which is existing on chromosome number 7. So on the left panel, you can see the 23 pairs of chromosomes that we all have, specifically within chromosome number 7. As we can see here, MET exists in this location right between, I think, what some of the discussion has been focused on, EGFR this morning and BRAF being another oncogene. And the role for this particular gene, it leads to expression of a protein, which, subsequently through phosphorylation is involved in recruitment of signaling proteins, and which are eventually and ultimately important for cell growth and cell signaling and repair. So MET is really like all other oncogenes. These are, genes, proteins, which we require for, cell functioning and repair, but essentially then become aberrant or altered, which lead to, formation of cancer. And in our context, formation of non-small cell lung cancer. So MET is quite unique because it has various mechanisms of alteration. If we think about it, it's not just one particular way that MET can be altered in a cancer cell, or it can be altered leading to cancer, but there's a variety of ways. So one of the ways is, point mutations, which is basically an error in the genetic code of the DNA, at one particular point. But then there is, MET amplification, which I think the easiest way to think about this is there's extra copies of that gene, rather than just having, that one copy, which can then lead to overexpression of that protein and then leads to over signaling and, proliferative growth leading to cancer. And then the third mechanism is this exon 14 skipping mutation, where you can, sort of think about this as there is an instruction which is missing in this particular sort of alteration, which then leads to that overexpression and overactivation of this pathway. So, but this is the skipping part of this is really, the missingness of this kind of domain, which is usually found in a normal gene. And then there's also MET fusion. We'll talk about fusions also in the context of NTRK and NRG1, but really MET is unique because that has multiple altered mechanisms, and it can get very confusing, even for us, because we talk about MET overexpression and alterations and exon 14 skipping and such. And In terms of the prevalence of some of these alterations that we see within the context of non-small cell lung cancer, MET exon 14 occurs in about 2 to 4% of non-small cell lung cancer as an oncogene driver. MET overexpression and amplification is much more common, or in a higher prevalence, and then MET fusion is quite rare. For the context of this discussion, I'm just going to be focusing on the MET exon 14 skipping part, especially because this is the area where we have FDA-approved medications or targeted therapies that we can use. There is an increasing body of literature and research ongoing for this particular aspect of things. I know we discussed a little bit about MET overexpression as a key resistance mechanism, in some of the more common oncogene-driven non-small cell lung cancer mutations. But for the context of this discussion on MET, I'm going to be focusing just on the exon 14 skipping portion. So as I mentioned earlier, this is just kind of showing how an exon 14 skipping mutation with an aberrant splicing, just being a fancy word for what I just said, is that missing instruction of the recipe. So with that, there is going to be the downstream pathway activation and decreased degradation of the protein, which leads to overexpression and, eventually leading to overactivation and carcinogenesis. And I think the important aspect here, especially with MET and some of the other fusion-driven non-small cell lung cancer oncogenes, and I think some of this discussion, again, kind of carrying over from was done in the previous session, is, there's a body of literature which suggests that the best way to identify, these mutations are through an RNA-based NGS analysis compared to just a DNA-based analysis. And this, figure here is really to show you some of the example of genomic alterations. And you can see that there are certain alterations which DNA-based tests will not be able to detect in MET compared to the RNA-based NGS, which is better for the fusion analysis with the fusion exon 13, 14, and some of the cases that I showed here. So I guess the take-home point is, again, and I know this was brought up, that you want to ask your oncologist on the kind of genetic testing you've had. Specifically, I would also encourage you to ask about whether the next generation sequencing assay that was ordered, is it a DNA-based only, or is it a DNA and an RNA-based NGS? And I think just, kind of going deeper into the, without getting too technical, but going deeper into kind of the actual, test that has been ordered so that you have a good understanding of what can and cannot be detected. So talking about how do we target MET, and there have been a variety of approaches that have been tried. So talking about sort of, if you think about this as a protein in the cell membrane, there's going to be some of the intracellular part and the extracellular part. The extracellular domains can be targeted by anti-MET antibodies. And I think we talked a little bit about amivantamab. I think that was mentioned as a MET and EGFR targeted antibody. But again, these are usually helpful in the MET amplification overexpression sort of state. For point mutations or exon 14 skipping mutations specifically, we have the selective MET inhibitors, which are truly targeting the overexpression of the protein and the aberrancy, which leads to the cancer. And so here again, I'm going to talk to you about, obviously there's been, as I mentioned, antibodies, and then we are going to talk specifically about the tyrosine kinase inhibitors, which have targeted MET exon 14. So the tyrosine kinase inhibitors are essentially drugs which can inhibit this sort of overactive, over-signaling aspect of the MET, oncogene-driven non-small cell lung cancer. And there's a variety of, names that you can see here, a variety of drugs that have been tested. And you see these Roman numerics right here. This is the different type of MET inhibitors. Essentially, the type 1B, specifically capmatinib and tepotinib, are the two MET TKIs which have been approved for MET exon 14 skipping non-small cell lung cancer. And here in this snippet, I'm showing you some of the data on the efficacy of these TKIs. So for example, for capmatinib, when it is used in the first-line setting, meaning once you have a diagnosis of MET exon 14 skipping non-small cell lung cancer, and if capmatinib is used as a first line of treatment, we have seen responses around 60 to 70%. This is essentially, 60 to 70% of patients that have been enrolled experiencing shrinkage of the tumor or stable disease or partial shrinkage of the tumor in the first-line setting. Similarly, tepotinib in the first-line setting has shown efficacy in the rate of 57 to 60% as well. And then as and when it becomes or is used in the second-line setting, we see that rate drop a little bit, when it's used in the second line. It has also been tested for MET amplification and MET fusion. Again, none of this is currently approved. We're just going to focus on the MET exon 14 skipping part of it. These are the two drugs that are approved by the FDA. There are other drugs on this list which may be approved in other countries such as China. But really, these are the drugs you will hear about when we're talking about MET exon 14 skipping mutation. There are other antibody drug conjugates, which I think were mentioned, which are again being studied in the overexpression state. And there are a few clinical trials targeting this. But largely right now in our treatment armamentarium, we have these two TKIs. So the management here, I'm showing, the snippet from the national guidelines, the NCCN guidelines. And again, here it is really to hammer home the point that it is really important to use targeted therapy for MET exon 14 skipping mutation, to the point that in the guidelines, you can see on the bottom here that if it is discovered during first-line systemic therapy, it may be recommended to interrupt the current therapy and start capmatinib or tepotinib as the preferred treatment. Obviously, if it is discovered prior to first-line systemic therapy, then the preference is to start capmatinib or tepotinib as first-line therapy. So really important to look for this mutation, and once detected, to target this mutation through one of these TKIs. In terms of safety, so these, again, as I mentioned, these are oral drugs. Capmatinib is given 400 milligram twice daily, and then tepotinib is given 400 milligram once daily with food. And the interesting part of, the safety profile is quite different from, some of the things that we are commonly using, because again, MET exon 14 occurs in about 1 to 2% of non-small cell lung cancer patients. It does have some of the common side effects. So for example, we have the GI disturbances, the increased liver enzymes that we have to monitor once we start a patient on this particular therapy, one of these therapies. Again, we measure the renal function for increased creatinine, but some of the specific side effects that we see with this MET TKI is peripheral edema. This can be asymptomatic, but can, again, depending on the grade, can become symptomatic. We monitor for skin erosions, and we consider prophylactic measures. We also consider dose reduction and interruption, as well as diuretics and corticosteroids as needed. ILD has also been seen with these TKIs. It's rare, but again, we are very cautious and monitor our patients for signs of interstitial lung disease, as well as pleural effusion in this particular, therapies. So now moving on to NTRK. So NTRK is neurotrophic tyrosine kinase receptor kinase. This again is a gene, and this is usually found to have NTRK fusion-driven non-small cell lung cancer. By that, it just essentially is that when you have an NTRK gene, NTRK 1, 2, or 3, and then when it sort of chromosomally translocates and crosses over and fuses with another gene in the chromosome, essentially leads to this oncogenic fusion protein, which again can precipitate uncontrolled growth leading to cancer. So again, this pictorial here shows that you have an NTRK gene, and then it can fuse with another gene through this process called translocation. And then once it fuses, it leads to this fusion protein, which can be an aberrant protein causing increased cell proliferation. And what's interesting with the NTRK is there is not just one partner that it prefers. It actually can fuse with many different genes that we have seen. And this is a picture. Again, this is Circo's plot showing all the chromosomes here, numbered from 1 to 22 with X and Y chromosome. And you can see that it can actually fuse with genes, pretty much across the spectrum with the NTRK. And that, once fused, can cause cancer. And NTRK fusion is not just found in non-small cell lung cancer, but also in many other cancers, specifically when it is driven through NTRK as the primary process. For NTRK fusion positive non-small cell lung cancer management, again, very important for us to detect this. Again, this is also preferably an RNA-based NGS, which would be important to detect this. Again, once we find this as a first-line therapy, we like to start targeted therapy. The therapies here are entrectinib, larotrectinib, or repotrectinib. And I know Dr. Bespina just talked about repotrectinib in the context of ROS1. It's also beneficial and effective in NTRK and was recently approved. Entrectinib and larotrectinib have been approved for the last couple of years. Again, if it is discovered during first-line treatment, it is recommended to interrupt current therapy and start. And this is obviously because of the efficacy that we have seen with these drugs. Repotrectinib was approved recently, as I mentioned, and it also has been indicated, especially in the guidelines, that if there is a progression on entrectinib or larotrectinib, could be utilized if not previously given. And this is the data on these three drugs. Again, you can see here the target genes are a little different. Entrectinib, repotrectinib also target ROS1 and ALK. And that's the reason we've seen repotrectinib in the context of ROS1 that was just mentioned. And then here you can see that the efficacy in terms of the overall response rates are in the order of about 60 to 70%. The treatment side effects, the profiles are slightly different. Especially with entrectinib, there are some CNS manifestations, but repotrectinib, as was mentioned by Dr. Bespina, there is also CNS-related side effects, specifically dizziness, that we see. We also see weight gain, and so some of the side effects are because of the targeting of this TRK A, B, and C, and ROS1 are quite similar, and also more related to the CNS as well as the new nerve system, just because of because the NTRK gene and protein actually is a neutrophilic protein that is very important for nervous system function. In terms of, and I know this has been covered in the morning, just talking about resistance. So again, here NTRK inhibitors, important to understand what the resistance is. This is again present only in 1 to 2% of non-small cell lung cancer is NTRK-driven non-small cell lung cancer. So we don't really have a lot of understanding of the different resistance mechanisms as clear as, at least as we understand EGFR or some of the more common oncogene-driven non-small cell lung cancer. But again, we know that there can be on-target resistance through solvent front, gatekeeper, and others, or as well as off-target resistance. So important when there is progression of the cancer to test either through liquid and, if possible, through tissue biopsy. In terms of safety, as I mentioned, there's ataxia, myalgia, peripheral sensory neuropathy, again, that nerve system-related side effects that we see, dizziness, dysgeusia, vomiting, weight gain, some of the common things that we can see with NTRK TKIs. Moving on to NRG1, which is the new Regulin-1. This serves as a ligand for HER3 or HER4. You may have heard about HER2 in the context of breast cancer. And so HER3, HER4 are sort of, receptors in the same family. And NRG1 actually is a substrate that binds to these receptors and then prompts the heterodimerization, and then leads to, again, downstream signaling cascades for cell proliferation and survival. So with the NRG1, we see that NRG1 alterations frequency is not just in non-small cell lung cancer, but even in pancreatic, renal cell, ovarian. About 0.3% of non-small cell lung cancer cases are NRG1 driven. Again, RNA-based sequencing is helpful, and we see that NRG1 alterations are enriched in invasive mucinous lung adenocarcinoma, which is a specific histology within the adenocarcinoma context for lung cancer. So if we see that, we want to think about NRG1, but again, the overall frequency is about 0.2 to 0.3%. So really, really rare to find this. But again, if we don't test, we won't find it. So interesting advances in this particular subset of non-small cell lung cancer in the past year or two. This is, I guess I would say, one of the few oncogene drivers where we want to use first-line treatment as sort of the standard chemoimmunotherapy type of approach. But then at progression of the cancer in the second line or subsequent therapies, we want to use a more targeted therapy. And in this context is zenocutuzumab, which I'm going to talk about briefly. But this is, again, the national guidelines and was also recently approved in December of last year. And so this has been the major change in this particular subset of non-small cell lung cancer. So Zeno is essentially a bispecific sort of mechanism with NRG as well as binding to this HER2. It blocks the NRG1 binding the HER3 domain and then inhibits that cell proliferation and survival cycle, which can be caused by that NRG1 alteration. As we know that NRG1 exists in multiple tumor types, so this drug was tested in multiple different tumor types, and again, it did show good response in non-small cell lung cancer in the order of 35 to 40% in the most updated data set, actually. And then also in pancreatic ductal adenocarcinoma, which is the other tumor type where this was approved also with the NRG1 alteration. So as I mentioned, it was December of last year where it was approved both for non-small cell lung cancer and pancreatic adenocarcinoma. It is notably an IV treatment, unlike some of the oral drugs that we talked about and were talked about in the previous session. This is every 2 weeks, and it has to be given along with steroids and antihistamines because it has infusion-related reactions that have been seen. So the major side effects or safety concerns that we want to be aware of are infusion-related reactions or hypersensitivity. Want to measure the ejection fraction, so do an echocardiogram of the heart to measure the pumping and assess the function of the heart. ILD is one of the other side effects that we want to watch out for. But the most common ones are diarrhea, musculoskeletal pain, fatigue, nausea, and the infusion-related reactions that we mentioned. So here are my key takeaways. MET exon 14 skipping non-small cell lung cancer, 1 to 2%. It is a targetable alteration. RNA-based NGS is important, as is for NTRK and NRG1. Capmatinib and tepotinib play an important role in treatment for MET exon 14 skipping. For NTRK fusion positive, we have 3 approved therapies, larotrectinib, entrectinib, and repotrectinib more recently. And then for NRG1, which is a very rare occurrence in non-small cell lung cancer in the order of 0.2 to 0.3%, but when present, responds to the zenocutuzumab, which is in second line and beyond setting. Thank you.
Thank you so much, Dr. Desai. We will not have time for questions. Thank you so much. We'll just go ahead and move to Dr. Nieva and to introduce Dr. Nieva. He is an associate professor of clinical medicine at the Keck School of Medicine in the University of Southern California. He's a member of USC Norris Comprehensive Cancer Center and a very good friend because we did fellowship together. He was 1 year senior than me, but I still remember that wonderful time. So go ahead and welcome Dr. Nieva. Oh, you're muted.
Thank you so much, Dr. Bazanova, and for giving away that somehow you've managed to age much slower than I have throughout this process. I want to thank Cancer Grace for inviting me to talk today on this very important topic of KRAS and BRAF. These are my disclosures. So KRAS and BRAF are a little bit different than all the other genes that you've heard about that are largely kinases. And a kinase that you've heard about earlier, they typically respond to some kind of signal on the cell surface. And in response to that signal, will then activate a protein just below there, and then transmit that signal basically from the outside of the cell to the inside. KRAS and BRAF are those second signals. So in the little cartoon that I'm showing you here, most of the things that we've been talking about so far are this pink molecule on the cell surface. And this pink molecule on the cell surface typically activates some kind of secondary protein that moves the message from the cell surface all the way down into the nucleus. Now, KRAS and BRAF are both very similar agents, very similar proteins, and they transition between an active and an inactive state. And in the process of doing that, they burn phosphorus and then activate a signaling cascade that goes down into the nucleus and has the cell start doing whatever it is that cell normally does. Now, many of you took high school biology and you remember mitosis. And mitosis, you'll recall, is basically cells dividing. And so these cells, in response to activated signaling, will divide. And they'll also do whatever it is that that cell is designed to do. So if it's a mucus-producing cell, it'll make mucus. And for many of our patients with lung cancer, that's one of the main functions that these cells do. So in addition to growing, these cells will also receive survival signals. So one of the things that cells have innately is that they recognize if things are kind of broken inside of them, if their DNA is broken, if their proteins are broken, they're supposed to die. And they'll turn on these programs called apoptosis programs to do that. But if they're constantly stimulated through these signaling pathways that KRAS and BRAF are such an integral part of, they won't listen to those other signals that are telling the cell to die. Cancer cells have many aberrant things about them, and there are many internal proteins that, if they were allowed to, would have the cell simply undergo a cycle of programmed death. But activation of KRAS or BRAF and the turning on of these pathways really prevents that from happening. And so patients who have mutations in KRAS or BRAF within their cancers, they will always have these growth and division signals active within them. Now, we've all been learning about how lung cancer is not one disease and how different signaling mechanisms really define lung cancer. KRAS and BRAF are largely adenocarcinomas, and we will see KRAS signaling in about 40% of adenocarcinomas, 25% of adenocarcinomas down here. And BRAF signaling, much less common, about 2% of the adenocarcinoma patients will have that. We don't really see very much KRAS or BRAF signaling in the squamous cell carcinomas. Now, unlike many of the gene aberrations that you've heard about up until this point, KRAS and BRAF are very frequently tobacco-related. And so these abnormalities are typically things that we see in people who've previously been smokers. Now, for KRAS, it's almost 100%. It's very rare to find a KRAS alteration in somebody who never smoked cigarettes. BRAF is different. BRAF is about 50-50. And we see some patients with BRAF alterations who are smokers and some who are not smokers. And these abnormalities can be present not just in lung cancer, but can be present in many different cancers of the body. The pancreas, the bladder, cervix, the mouth, tongue, stomach. So you really don't want to smoke if you can avoid it. But if you do currently smoke, your chances of getting cancer actually comes down within 2 years. And now most people who try to stop smoking will be unsuccessful. In fact, people are only successful when they stop smoking um, uh, each attempt about 7% of the time. So it's super, super hard to quit. The good news is that you can just keep trying again. And so people get discouraged because they say, oh, I tried and it didn't work. And then I tried again and it didn't work. But the beautiful thing is that you can just keep trying. It never has to stop, and you never have to stop trying. And eventually, just about everybody eventually can quit. And that's the great news. And then one of the other things to keep in mind is that it's much better to be on some kind of nicotine replacement rather than smoking. So, there's a lot of nicotine replacement products out there, gums, patches, even vapes. They're all better than smoking from the standpoint of cancer risk. So when people are smoking, it leads to a lot of damage in DNA, and it can create these alterations in both KRAS and BRAF. But those aren't the only alterations that are created. One of the things that we find is that smoking leads to a very high, what we call tumor mutational burden. And you may have remember people talking about this in the earlier trials of immune therapy. Tumor mutational burden is a measurement of how many mutations exist in DNA for every 1,000 DNA base pairs. And we consider someone as having a lot of mutations when there are more than 10 per 1,000 base pairs of DNA, or 10 per megabase. And so when you look at people who are smokers and you look at how altered the DNA is in their respiratory epithelium, you'll see that it's quite abnormal for the majority of smokers, and that's the column here that you see in red. But as you get further and further out from smoking, you see that the mutational burden gets better gradually, not immediately, but gradually, and begins to get closer to the mutational burden that we'll typically see in a lifelong non-smoker. So we can take advantage of these DNA aberrations in people with BRAF and KRAS alterations by using the checkpoint inhibitors that you've heard about earlier today. The way these checkpoint inhibitors work, and I'll just to remind you, is they make it so that the immune system better responds to broken proteins. So this abnormal DNA translates to abnormal proteins. And those abnormal proteins look not like yourself. They look like they came from something that's not you, because your body's immune system recognizes all of your own proteins from birth, essentially. So these proteins that are inside of cells are constantly being exposed to immune cells through a series of immune regulatory proteins called the MHC proteins. And you see here a cartoon on the right of an internal protein, an internal antigen of the cell being presented to an immune cell. And essentially what's happening here is every cell in the body is constantly showing its inside out and is taking internal proteins and displaying them on their outer surface. And as immune cells come by and interact with that cell, they kind of check and have sort of an inspection process. And they check to see if any of those proteins are foreign. And if they're not foreign, if they've been with you all your life, no immune cell will actually ever react to it. Immune cells go through a process where if while they're growing up, they react to anything inside your body, they actually get destroyed. So all the immune cells in your body actually don't react to any of your own cellular protein contents. But if there's a protein inside of you that's abnormal, well, then your immune system will respond to it and actually will then want to destroy that cell. Now that process is one that can be regulated and made stronger by these immune checkpoint inhibitors. And what the immune checkpoint inhibitors do is they make it so that very weak interactions against proteins that are only slightly abnormal become triggering interactions and make the immune system destroy that cell. So immunotherapy really is the mainstay for our tobacco-related cancers and lung cancer. And so that's KRAS, and that's about half of the BRAF patients, the ones with tobacco exposure. And you can see here Kaplan-Meier plots, and I'm sure you've been looking at Kaplan-Meier plots all day. And what's really terrific about what immune checkpoint inhibitors like pembrolizumab do is they create this flat part on the survival curve. These are people who look like they're going to be long-term survivors, even though they had stage 4 disease. Really a miraculous outcome when you think about where we were with lung cancer 10 to 20 years ago. Now, immune checkpoint inhibitors work well, but there are certain situations where they're going to work not so well. And so it's important when you're looking at people with KRAS or BRAF alterations that you look to see whether they co-express these other proteins that can inform whether or not immune checkpoint inhibitors are going to work. So when you look at a next generation sequencing report, you'll want to pay attention to the status of a gene called KEAP1, NFE2, and STK11. The presence of any of these genes, abnormalities, mutations in these genes in people with lung cancer and KRAS or BRAF alterations makes the immune checkpoint inhibitors less likely to work. Also, if the cancer doesn't express any PD-L1 on its surface, they're less likely to work. But if the tumor mutational burden is particularly high, if it's 20, 30 or more, they're actually more likely to work. And so we can kind of get a sense of which patients these drugs are going to be better for and which ones they're going to be perhaps not as good for based on these characteristics. Now for KRAS and BRAF, we now have targeted therapies. KRAS was often considered undruggable, and it was considered undruggable because if you inhibited the RAS protein in a mammal or a human, then all the cell signaling would stop in all the cells of the body, and all the cells would die. Effectively, would be like a poison. But the folks at Amgen and a variety of other pharmaceutical companies now have developed drugs that only inhibit RAS if there's a mutation in RAS. So only if it's a broken RAS will it be inhibited. And if it's a normal RAS, it won't be inhibited very much at all. And these drugs really generate remarkable responses. And it's just like what you see with all of the other very exciting oral tyrosine kinase inhibitors in all the other presentations, where we see the vast majority of patients seeing their tumor shrink. And so here I'm showing you data from sotorasib and adagrasib, which were the first 2 drugs that came to the market and are the currently 2 only FDA-approved drugs, where you see most people's tumors getting smaller rather than bigger once they start the drug, and that's what these waterfall plots show. The target has now been evaluated by a number of other pharmaceutical companies, and we're now seeing mutation-specific inhibitors from lots of other folks. Now, because they're mutation-specific, they only work against a very specific KRAS mutation. So in the case of sotorasib and adagrasib, they don't work against all KRAS mutations, only the KRAS G12C mutation. So it's a change in amino acid number 12 from a glycine to a cysteine. Only if that's the mutation that caused the KRAS to be abnormal will these drugs work. Now, there's other companies working on inhibitors of KRAS G12D. There's other people who have inhibitors that work against lots of different mutations. So they work against more than one mutation. These are all going to be new developments that are going to be very exciting. Now, the thing is, these two drugs don't mix well with immunotherapy. And the combination of the two can be problematic because people can get liver inflammation from these drugs, and they can get liver inflammation from immunotherapy also. And so when you mix them together, they just don't mix well. There are people who are working on drugs that don't cause as much liver problems that may be mixed better with immunotherapy. And so we're really looking forward to seeing offerings from other pharmaceutical companies in the future that have that characteristic. Now, for people with BRAF alterations, there's actually a couple of different FDA-approved combinations. The thing with BRAF that's kind of funny is if you just inhibit BRAF, a compensatory signaling pathway in a gene called MEK will turn way up, and the patients will actually start to develop growths of cells kind of on cell surfaces, on their skin and other places in the lining of their airways. That's problematic. So you don't want to inhibit BRAF by itself. You always have to inhibit MEK every time you inhibit BRAF. And this means that when you're taking a BRAF inhibitor, you're also taking a MEK inhibitor. Now, the first combination of BRAF and MEK inhibitors that were approved in lung cancer were the combination of dabrafenib and trametinib. There's also another combination that's been more recently approved, encorafenib and binimetinib. And they work very similarly, a lot like Coca-Cola and Pepsi-Cola. They're more alike than they are different. But here, again, you see a lot of patients who take this combination very profoundly see their tumor shrinking. Now, the biggest problem with this combination, of course, is it causes fever. It's kind of weird, but that's what it does. And that's troublesome to patients. People don't feel well when they have fever. So they actually oftentimes have to take Tylenol just around the clock 3 times a day to suppress fevers. And it can be a little scary at first because people are used to when they're having fevers thinking that they might have an infection. But these fevers aren't caused by an infection. It's just that the drugs change your body's temperature set point, make you hot, and run a little hotter than what you would otherwise run. And that's really, I think, the biggest management challenge for people on these drugs. So just to review the key differences between these drugs, KRAS, about 25% of lung adenocarcinomas, BRAF, about 2%. KRAS, almost always smoking related. BRAF is about 50-50. For KRAS, any targeted therapy that we use has to be mutation specific because you can't just go around inhibiting all the KRAS in all the cells of the body. That just doesn't work. Whereas with BRAF, you can inhibit BRAF, but you also have to inhibit MEK. And if you don't do that, then you get these kind of weird, funny growths on the skin and other mucosal surfaces. Both of them respond to immunotherapy, but with BRAF, you're more likely to respond to immunotherapy if you used to be a smoker. If you have a BRAF mutation and you never smoked, immunotherapy is much less likely to work for you. So to summarize, these are molecules that transmit signals from the outside to the inside. These are very common. They're tobacco-related often. But immunotherapy is really the mainstay of treatment for these patients, particularly in BRAF if they were smokers. But the good news is we have targeted therapy now for those times when immunotherapy doesn't work. And in the future, I think we're going to be able to mix these drugs with immunotherapy as newer agents get developed. Thank you very much.
Thank you, Dr. Nieva. We are running a little bit over time, but I'm going to ask you one question. The discussions about BRAF that you had and the drugs that you mentioned approved for a very specific BRAF mutation called BRAF V600E. What do we do with patients who have a BRAF non-V600E mutations nowadays?
Well, in the year 2025, we treat them with immunotherapy or we treat them with chemotherapy. So you're correct that they really are only for that specific V600E mutation. That's the mutation that they really bind to well. The good news is there are going to be drugs coming for what we call the class 2 and the class 3 BRAF mutations. These are mutations in kind of different parts of the molecule. And so those parts of the molecule have different interfaces with other proteins. And so they need to be targeted slightly differently.
Perfect. Thank you so much. Thank you, Dr. Nieva. And it is my pleasure to introduce our last speaker of the day, Dr. Huning Lee. She is an associate professor at the Department of Thoracic Head and Neck Medical Oncologist at MD Anderson Cancer Center, and she will take us through EGFR and HER2 targeted therapies. Go ahead.
Yeah, thank you, Ludmila, for giving us the opportunity, and then thank you, Grace, for providing this platform. Really, it's a fantastic time to discuss different type of lung cancers. Now, we just heard about KRAS and then BRAF. I'm going to be talking about EGFR and HER2. Also, there are two associated targets very prevalent in lung cancer patients. Those are my disclosures. So today I'm trying to cover ground mostly on the frontline treatment because my colleague, Dr. Eric Singhi, was covering the resistant situation. So I'm going to focus on frontline treatment for EGFR, including classical mutation, exon 20 and PEG, and we'll be talking about some of the new development in HER2. EGFR lung cancer now can be classified into 4 different groups, including classical mutation, T790M. Those mutations can be interrogated by treating with a 3rd generation EGFR TKI. In addition, we identified 2 structure function group. One is called PEG mutation, the other is called exon 20 mutation. Currently, we have a different exon 20 mutation approval I will be discussing, and then PEG mutation, we also start to see new clinical development, potentially new drugs gonna enter the clinic in 2025. Starting with classical mutation, we as a society has been using Tagrisso or the drug name osimertinib for the last 5 years or so based on the FLORA trial. In that trial, osimertinib frontline treatment for newly diagnosed metastatic EGFR patients achieved a progression-free survival of 18.9 months and overall survival 38 months superior than sequential use of the 1st and the 3rd generation EGFR TKI. In the last 2 years or so, we started to see and start to use FLORA-2, which is osimertinib with chemo combination in the frontline setting. And then we also have FDA approval of amivantamab with lazertinib in the frontline setting. So each of the trials I will go over today, discussing their superiority to the monotherapy osimertinib and also discuss what can be the potential problems associated with additional intensification. This is the trial design for FLORA-2. The trial enrolled about 550 patients and then randomized them to osimertinib with chemotherapy compared to Tagrisso by itself. This is their primary endpoint, showing that the progression-free survival is significantly superior when osimertinib is combined with chemotherapy, both by the investigator assessment and also by the RECIST assessment. In the subgroup analysis, for patients who have EGFR mutation lung cancer upfront have diagnosis of brain metastasis, this chemo plus osimertinib regimen confer significant clinical benefit with a hazard ratio of 0.47. Of course, for patients without brain metastasis, we still see benefit. However, the most significant benefit come from CNS metastasis patient population. This is another busy slide showing additional subgroup analysis. I boxed some of the significant findings in the red box, meaning that EGFR A58R mutation or detectable ctDNA at baseline or TP53 co-mutation, those high risk factors are associated with additional benefit with the use of chemo plus osimertinib. So with this subgroup analysis information in clinic, we start to discuss more with our patients when we see one or two or even more of those risk factors and then thinking through the risk and the benefit. Up to today, we do not have the overall survival data, but last update of the immature overall survival start to see a separation. Potentially, this regimen can also give patient overall survival. Hopefully, the data will mature in 2025. Around the same time that Mariposa trial also is a frontline EGFR mutation study reported positive results. Here, instead of using chemotherapy as an intensification regimen, amivantamab, which is a bispecific antibody, was used as the intensification regimen. So amivantamab with lazertinib, which is also a third-generation EGFR TKI, was offered, and then those groups are randomized to compare to osimertinib monotherapy or lazertinib monotherapy. The total enrollment of this trial is over 1,000 patients. The primary endpoint similarly is progression-free survival, and we also see significant benefit in the amivantamab plus lazertinib arm. In their subgroup analysis, brain metastasis patients also have additional benefit with hazard ratio of 0.69, as well as patients without brain metastasis. In their subgroup analysis, they included patients with liver metastasis here that amivantamab showed significant benefit for patients who have liver metastasis. One discussion point I usually have with my patients is the toxicity of amivantamab with lazertinib in comparison to osimertinib monotherapy, as well as in comparison to OC plus chemotherapy. In this relatively new regimen of AMI plus laser, we start to see grade 3 or higher toxicities around 75%, including skin rash, including hematological toxicities, as well as a DVT risk increase. With that, amivantamab team created a subcutaneous form and reported out a PALOMA-3. Although this formulation is not currently approved in the United States, but subcutaneous amivantamab can remove some of the infusion reaction problems, as well as some of the anticoagulation needs. Furthermore, when we compare the results, we have to say that a couple of weeks earlier, overall survival data was reported to be positive for Mariposa trial. By press release, the data will be released next week at ELCC. So we look forward to see how we are changing the landscape and then moving the intensification regimen up front to improve our patient survival. Switching gear a little bit, we'll talk about EGFR exon 20 mutation. EGFR 20 insertion mutations are different than classical mutation because those insertions create a slightly smaller ATP binding pocket so that osimertinib, for example, doesn't fit into this ATP binding pocket, so doesn't work for this type of lung cancer. Amivantamab as an antibody drug was able to overcome that problem, and in the Chrysalis trial, one arm of the amivantamab monotherapy showed a significant benefit with response rate of 40%. Therefore, is approved by US FDA to be used in the second-line setting. About 2 years ago, a Populon trial reported that amivantamab with chemotherapy compared to chemotherapy as a frontline treatment option is superior, so that currently we're using amivantamab with chemotherapy as a frontline treatment for EGFR exon 20 mutation. This is a progression-free survival at 11.4 months compared to chemotherapy, and the response is also higher with ami and chemo. Although currently we don't have an oral formulation or a targeted therapy for EGFR exon 20, there are a number of drugs that's on the horizon potentially to be FDA approved in 2025. Currently, there are 3 drugs under FDA with FDA breakthrough designation. That's savolitinib, zipalertinib, and furmonertinib. All 3 drugs are under phase 3 evaluation for the first-line EGFR exon 20 studies. Hopefully, we will be able to offer oral medication for EGFR exon 20 patients, and then subsequently use amivantamab as the subsequent IV treatment option. Last but not least, PAC mutation is a newly recognized subgroup, mostly in the EGFR exon 18 spot. We start to have furmonertinib and other targeted therapy enter into this space. This is a report that we had seen last year, reporting furmonertinib as a efficacious medication, especially at high dose with 100% disease control and 80% best response. So we are also looking forward for PAC mutation patients having a TKI as a treatment option. I will skip the resistant part as this one was covered by my colleague, and then due to the interest time, I'm wanting to move into the HER2 space. So HER2 have a number of different names. It's called HER2. It's also called ERBB2. It's actually a sibling of EGFR, as they have a very similar structure function as transmembrane domain receptor. HER2 mutation occurs in around 2 to 3% of non-small cell lung cancer patients, and with YVMA to be the most common insertion mutation. We have in HER2 currently under FDA approval for second-line treatment. However, the last couple of years, the development focus has been developing HER2 small molecule inhibitors to the frontline setting. There are 2 medications, BER-88 and zongertinib, both had excellent preclinical data, and then they entered clinical evaluation. Last year, we reported BER-88's data for previously treated HER2 non-small cell lung cancer patients. As you can see, this TKI is highly efficacious with a response rate of 72%. Similarly, zongertinib also entered their phase 1 study, showing similar or potentially even superior benefit of response rate approaching 70%, and then the progression-free survival at the time of report not being reached. The toxicity of those 2 drugs are similar to what we know about the EGFR and the HER2 on-target toxicity. BER-88 have a little more diarrhea, and zongertinib has a little higher liver function toxicities. Currently, we're hoping to bring BER-88 and then zongertinib at least first in the second-line setting to give our patient additional option. And in the meantime, there are global trials ongoing evaluating either of the drug in the frontline setting in comparison to chemoimmunotherapy. Both of the trials' primary endpoint is progression-free survival. With that, I will conclude the HER2 part. Currently, we have in HER2 as the only targeted therapy, although it's an IV form. we are, as a field, developing rapidly novel oral medications, potentially in first-line setting, to add additional options for HER2 patients and prolong our progression-free survival. Thank you.
Thank you so much. Thank you, Dr. Lee. We actually caught up a little bit, so which is good. I will have a couple of questions for you. The one question that I think we really have not talked about, so I think right now it's a perfect opportunity to answer. So when you were talking about HER2 mutations, you were talking about antibody drug conjugate. And the question is from the audience, if antibody drug conjugate works on one type of mutation, why don't they work on all of them? What is the magic with the HER2 ADC that allows it to work against HER2 mutations?
So in HER2, the antibody drug conjugates, in theory, should work for all HER2 mutations. Currently, the label is non-small cell lung cancer with mutations of HER2. They didn't specify what type of HER2 mutation. But I can imagine that some of the transmembrane domain mutation, for example, in the 310 region, might not work as well because those mutations might not result in upregulation of the signaling and the cell surface expression of HER2 receptor. On the other hand, targeted therapy, small molecule, they're going to be very selective in terms of type of mutation. So we look forward to see more data on the translational front for BER88 and zongertinib.
Thank you. And I think I'll do one last question. I think a lot of questions that we have in there, you already touched upon during your discussion, so I'm not going to repeat them. But one question that I just got put in, which I think we deserve to discuss, what is the latest research and development on patients with EGFR mutation, which is co-mutated with P53 and RB1? Are you approaching those patients differently at the onset of treatment?
Yeah, so this is a challenging scenario we've recognized as a field for probably 2 decades, initially described by Dr. Alicia Seacrest. So when EGFR mutation co-occur with TP53 and loss of RB1, the risk of transform to a small cell or neuroendocrine is high. However, we still don't have a solution as a field to what to do, especially at the initial diagnosis. We know what's not working, so we have trials trying to prevent the transformation by offering patients with etoposide-based chemotherapy actually didn't result in additional benefit. Currently, for small cell lung cancer, we have made significantly progress, including DLL3 targeting. For tumors after small cell transformation, if the tumor expresses DLL3, I think it's reasonable for those patients to be treated with DLL3 targeting approach. In fact, at MD Anderson Cancer Center, we have a couple of successful cases. But how to prevent the transformation, I think, is still a very active research topic and area. A lot of us are putting significant effort into it.
Thank you so much. And with that, we will conclude the today presentation. I'd like to thank the program sponsors, which were Bristol Myers Squibb, Eli Lilly, Genentech, Merck, Daiichi Sankyo, Takeda, and AstraZeneca. For the audience, please fill out our post-event questionnaire with your feedback about the event. You can find that questionnaire by clicking the event questionnaire link at the bottom left of your current screen, or we also will be sending the link in a post-event mail. And again, this will conclude our GRACE 10th annual targeted therapy event. And I'd like to thank all the presenters. And I'd like the presenters to, after we discontinue the seminar, to stay for like 1 more minute to show on a camera so we can take a picture of the event. And thank you very much. Bye.